So, welcome everybody. I think that because we have quite a busy agenda, we have to start in time. I don't know how far we are all here, but maybe because of security measures, somebody will join us later. Some of you know me, but for those who don't, allow me just to introduce myself. My name is Pavel Poc. I'm a member of European Parliament from Socialist Group. I'm Vice Chair of Environment Committee, Chairman of Climate Change, Biodiversity and Sustainable Development Intergroup of the European Parliament, and Vice Chair of Members Against Cancer Group. And I'm very happy to host and chair this meeting on glyphosate. And as a chair, I have to give you a few organizational notes. First of all, big thanks to our organizing partners, the European Association for Chemical and Molecular Sciences and the European Chemistry Thematic Network Association for working with us and making this truly scientific conference happen. Another big thanks goes to all our speakers for making the time to participate in this event. Last but not least, I would like to thank all the participants for joining us. I believe it will be an interesting conference for you. And of course, I would like to welcome my honourable colleagues from European Parliament here, especially my colleague from Environment Committee, Julie Gerling. Welcome. Our conference is being recorded by automated video system of European Parliament, so in order for the cameras to work properly, I would like to ask everybody in the room to turn your micro off when you are done with speaking. The video will be, of course, available after the event and shared as soon as possible with you. Then I need to inform you that there are journalists of the German television ARD in the room over there. They are recording bits of the meeting as they are preparing a documentary on glyphosate for German TV. I hope that it doesn't mind you. We are limited on time, so in order to keep time for discussion, I have to ask all speakers to really stick to allocated time, and I will dare inform you when your time is about to run out and ask you to conclude your speech. And at the last resort, I have this hammer to first I will do this and then I will do something else. After the first session, we will have a short coffee break at around 4 p.m. And on your table there should be an agenda. I hope everywhere it is. The case conference brochure including every speaker's bio and presentation abstract, and also a small gift bag. So now I would like to inform you about a few things. First, how this event was born, then on my relation to the topic, and then last developments here in the Parliament on Monsanto paper, and then I will give the floor to first of our speakers. So, on how this event was born, I have to thank cordially to Professor Pavel Drashar, who I met first in November 2016 in the frame of MEP Scientist Pairing Scheme, which is organised by European Parliament. In this programme, scientists are shadowing our work in Parliament, with the aim to understand each other's work and find new cooperation channels. And in this case, it really worked, and Pavel gave me a new impulse, which I needed, in things where I was not too far from giving up, and it was a case of glyphosate. We have written this nice paper together, and so we started to prepare this event as an answer to the demand to deal with glyphosate agenda in, say, more scientific-based manner. So this event really was planned and prepared quite a long time, and only by chance currently came Monsanto papers, which probably gives more spicy taste to our debate. On me and how I am joined with this topic, I was dealing with some health issues in my first mandate, especially digestive cancer. After being scared by the scale of this problem in the European Union, I decided to make my priority in this mandate conjunctions between our health and the chemical pollution of our environment. This statement sounds corny, I know, but when it comes to public governance, legislative, industrial, or even daily life decision-making, health is often, if not mostly, outvoiced by other interests. Money, it is about money. I want to shift this disbalance back to the common sense as far as it will be possible. And among other steps I have tabled in this regard also objection against longest possible unconditional reauthorization of glyphosate after contradictions between EFSA and IARC in 2015. At the end of the process, there was an EP, European Parliament resolution appealing to the European Commission to authorise the compound not longer than 7 years, to ban non-professional use, to ban use on public grounds, to ban using of this compound for pre-harvest treatment, etc., etc. After Member States did not support the Commission proposal, European Commission decided to prolong the former authorisation for 18 months and to put glyphosate under the survey of ECHA. This period ends soon, and ECHA apparently decided to give to glyphosate green light. We will hear why today. In the meantime, I have read a lot of studies about glyphosate, discussed with many former colleagues, scientists, my former colleagues and friends. And as a physiologist by education, I had a chance to get clear enough picture about how this compound can interfere with our cell chemistry, how deep into the metabolism it can encroach. And you will hear most important things today from those who are dealing with this issue. Last remark is on Monsanto papers. I don't think it is necessary to elaborate on this too much. If you are here, that means that you are interested and you are informed. So very briefly, internal Monsanto emails were released by US Federal Court of San Francisco earlier this year. This communication revealed that the company could be involved in ghost writing scientific studies on glyphosate that were later attributed to some scientists with the intention to influence assessment of the substance and probably cast doubts on conclusions of IARC from 2015. I think that I should probably inform you about what has to be done on the EU level and what I will impose in my political group. And it is 8 points. In case of Monsanto influence or any other company influence on the reauthorization of glyphosate, the Commission has to adopt any necessary legal steps immediately. No delay is acceptable. It is absolutely essential that any attempt to manipulate scientific evidence to influence the EU authorisation processes are duly treated and thoroughly investigated. Oral question with resolution will be used as the first immediate step to develop pressure to the Commission and the Council in this regard. Joint ENVI Committee and Agri Committee hearing on the issue has to be organised with the relevant stakeholders as soon as possible. If there are any doubts, then Parliamentary Inquiry Committee has to be established to find out how Monsanto or any other subject could have impaired the decision-making process and on which level it could have happened. In the EU, pesticide authorisation procedures are possibly disrupted. We have to reveal how such an influence could have happened and close possible gaps. Non-such investigation has to be understood as undermining of our trust to the agencies, but improvement of such trust in case it can be or is endangered. Glyphosate, by my opinion, is a good example of how fragile is our system, and we have to use it to improve pesticide legislation, to improve our authorisation procedures with only aim to maintain and improve our food safety and the same way to maintain and improve our citizens' health protection. A last remark is about that I was established as a rapporteur for ENVI Committee implementation report on the plant protection product regulation this week. Of course, I am aware about the limitation of this procedure, but I hope even this report could be used to propose necessary improvements of our regulations and authorisations procedures. And now I am happy to give the floor to Mrs. Pilar Goya, EuCheMS Vice President.
Thank you very much. Good afternoon, ladies Ladies and gentlemen, and thank you very much for coming to this very important workshop in the European Parliament. I would like to start by thanking Pavel Poc and also Professor Pavel Drascher, who are paired through the MEP Scientist Pairing Scheme, for bringing us all together. And I would also like to thank Ninetta Harstel, Bruno Vilela and Marta Kucha for organizing things on behalf of the EuCheMS. The meeting is being held jointly between the European Chemistry Thematic Network, which is chaired by Pavel Drascher, and the European Association for Chemical and Molecular Sciences, EuCheMS, of which I am President-elect. The President, David Cole-Hamilton, sends his apologies for not being able to be here today. As some of you may know, EuCheMS coordinates the work on all the chemistry departments in Europe, and as such represents an independent and unbiased voice on all matters to do with chemistry. For this reason, the meeting this afternoon will concentrate only on the scientific facts concerning glyphosate. Glyphosate has been extensively used since the 1970s, but however, as you have just heard, there have been reports that glyphosate may have adverse effects in humans, including being carcinogenic, but there is considerable controversy still today, and we shall look today at the scientific evidence that is available. Glyphosate is used in a variety of formulations, so there is also a debate whether, if there are any adverse effects, this may be due to the glyphosate itself or to other substances which are added to the formulations. All of these issues will be discussed in this meeting, with the scientific evidence being presented by a variety of international experts working in different disciplines. The meeting is highly timely, as the European Chemical Agency, the ECHA, reported its opinion on March 2017 that the available scientific evidence did not meet the criteria to classify glyphosate as a carcinogen, as a mutagen, or as toxic for reproduction. The European Commission, as you have just heard, must decide whether glyphosate can be used in Europe or not. Let me just finish by thanking all the speakers for agreeing to contribute to what I think is going to be an excellent meeting, and I very much hope that we will all learn a lot from it. Thank you very much.
Thank you very much as well. Thank you for it too. Yeah, okay. Very good. It was for both, I hope. And now I'm very happy to give the floor to scientists, my friend now, professor in University of Chemical Technology in Prague, ECTN president and EuCheMS executive board member, Pavel Drascher, to elaborate on current controversy in the case of glyphosate. The floor is yours.
Thank you very much. I volunteered to say a few words at the beginning of this important discussion. And first, let me start with the pointing out that this world is nowadays very much influenced by a phenomenon called chemophobia. People, or let's say general public, refuse everything where is some chemistry. You can even see it written on some products, and as I'm stating on a slide, it's sometimes very difficult to convince a physician that E300 is vitamin C, especially when he sees its chemical name. There is famous Professor James Kennedy in Australia, now teaching in China, who tries to fight the chemophobia. And he publishes posters like this when he shows what's the content of our food, our cosmetics, and our daily products. And if you see all these chemicals in normal banana, and you are a chemophobic person, you probably won't touch it even with a wooden stick. And But today we are here to talk a little bit about other phases of chemistry. We have many chemicals. They are used in agriculture, and one of them is Roundup or glyphosate. And if you can see that the use of, and this is the active ingredients, not the packaged product. If you see that there is 0.5 kilogram of glyphosate of every hectare of arable land in last year, not last year, 2014, and you see all the consumption which goes to tens of billions of tons of chemical, and simultaneously, when you have a Roundup ready crops like soya or maize, and taking into the account that nowadays most of the glyphosate goes from China. And my personal experience is that if I buy a chemical from China, it's sometimes cheaper than in other parts of the world. But if I am lucky, I will get product where there are 40% of the compound desired. So what is in the product which goes from China is sometimes very questionable, and sometimes it may raise also big concerns. Some time ago, the International Agency for Research on Cancer, not having enough information on human carcinogenicity, reclassified glyphosate as probably cancerogenic for humans. There were enough of evidence, I mean scientific evidence. The problem is that when the people made a survey on the glyphosate literature, so the majority of the literature was not from the, I would say, scientific journals with the peer review. Those were public informations like some pages on web without any validation and without any scientific value. So this evidence is sometimes taken, and as you see from the material of Monsanto, they say that despite of the overwhelming weight of evidence that the glyphosate is harmless, they do not agree with IARC's comments. On the other hand, you have on TV, and this is still present on YouTube, a film which shows a lobbyist who says that he can drink a glass of Roundup without any problems, and when he is offered a glass with let's say, sort of a liquid, he refuses. So we are having problem. And when we saw with Pavel that glyphosate really acts against cytochrome P450, it is something for me as a natural product chemist, something really striking, as the P450 is responsible for all steroid metabolisms in a human body. It's responsible for detoxification of toxic substances and for many, many other actions. And when this is true, we were really concerned that at least a discussion should be provoked. And when I also see that the glyphosate has a, I would say, very small, but still a structural similarity with the chemical warfare called VX, I am concerned once more. I should say that on one side there is a there is a necessity of politicians and all the humans to take care about the crops, to take care about the starving people, and where we still have many people on the globe, they are starving, despite the genetically modified crops and despite the pesticides and so on. We should also take into the consideration that the pesticides kill 200,000 people a year. So on the question of scientists, if it is really high time to release current safety standards on glyphosate-based herbicide, I say yes, it's the high time we did something. And for the reason, I wish you successful workshop.
Very well. Thank you, Pavel. I have two things to tell you. First, I'm a gardener, and it is a fate of all of us to have a cold in the spring. So I'm very sorry for running out. I mean, because I have really cold. So maybe Pavel can replace me sometimes when I'm,
Yes, I will.
not able to continue as a chair. I'm happy to welcome other members of European Parliament. Stuart Agnew, hi, hello. Jan Huitema, who just went out, was here but just went out. Frank Bogovic, hello, and Michel Rivasi, welcome, and I'm happy to have you here. Our next speaker, Tim Baumer from European Chemical Agency, is chair of Risk Assessment Committee. I'm especially happy to have him here because this Risk Assessment Committee assessed glyphosate, and he probably will tell us about conclusions and how they came to these conclusions. The floor is yours.
Thank you very much, Pavel. Pleasure to be here to speak to you, but also because it's snowing in Helsinki and 0 degrees today, so it's nice to find some spring for a change. My name is Tim Baumer, as just introduced. I'm chairman of the ECHA Committee for Risk Assessment. This presentation is quite technical. Some of the slides are quite busy. I will try and get through it in reasonable time, but I would like you to understand the background and some of the detail to our recommendation. Briefly about the agency, established 10 years ago. We have 600 staff from around the EU and the EEA countries in Helsinki. We are responsible for the implementation of the REACH regulation, classification, label and packaging, biocidal products regulation, and prior informed consent. Our main activities are to manage the 4 regulatory processes I've just described and deliver opinions and decisions. We also disseminate information on chemicals and develop scientific IT tools. We have a lot of communication work, as you would expect an EU agency to have. Talking about my committee, ECHA has 4 of them. Permanent committees. Most of these are devised and implemented in the various regulations they serve, as is RAC. It's composed of independent experts nominated by the EU or EEA countries and appointed by the ECHA Board of Management in their personal capacity. So they don't represent their member states. They're there as scientists. Currently has 51 members from about 26 member states. It varies from month to month. They're there on a 3-year renewable term, and their member states guarantee at least 50% of their time is spent on committee activities. A little bit about transparency. We have eligibility criteria in place for membership. Members are screened upon nomination by the member state. They're not allowed to be employed in a private capacity by industry associations or any other body with a direct interest in our work. They are obliged to sign annual declarations of interest, as, by the way, are ECHA staff. Member states are not allowed to brief committee members. That's in the REACH regulation, but they are obliged to support them in their work. It's an unusual formula, but it's one that works for ECHA. In terms of contribution from outside stakeholders, we have 103 accredited stakeholders representing industry, academia, and civil society. RAC meetings are attended by 7 regular stakeholders, including EuCheMS, I have to say. Okay, let's talk about the legislation and what classification and labelling means for a minute. In the flow diagram, you can see a whole process of taking a dossier through harmonized classification and labelling. This leads to a classification and labelling which is implemented across the EU and applies in transport and in all sorts of other downstream legislation. So CLP feeds into pesticides, biocides, the Water Framework Directive, transport regulation, and a lot of other areas. It's a piece of horizontal regulation with quite a severe impact on chemicals in general. When a dossier comes in, it's registered in the Registry of Intent so that everybody can see that it's there and about to be discussed. A dossier submitter is usually a member state. It can be industry under limited circumstances, but usually a member state submits a dossier to ECHA. That dossier goes through an accordance check, so it's looked at to see if all the bits are there. And then as soon as it's agreed to be in accordance by ECHA, it's put out to public consultation for 6 weeks. So the whole dossier is put to public consultation. We then gather the information from that. It can be almost nothing, or it can be up to, in some cases, 1,000 comments which have to be sifted through. A response to comments document is prepared. That is with the help of the member state submitting the dossier. And when all that is taken care of, when all the public contributions have been added to the dossier submitter's proposal, it gets to RAC. In the case of glyphosate, we held a key issues consultation in December last year. We did that deliberately with a range of outside agencies. I'll list them in a minute. Then it goes into the development of the opinion. Rapporteurs are appointed from the committee, and it goes through the rest of the process. Once we have finished, and we have a maximum of 18 months to our legal deadline, the recommendation is then in the form of an opinion passed on to the Commission for a decision. I'd like to talk about weight of evidence in the legislation. CLP is, in fact, has a long history. It goes back into the Dangerous Substances Directive to almost 50 years ago. It was updated in 2008 and modernized. It's harmonized with the globally harmonized system of the UN. We have to sift evidence when we get it in a dossier. And in the case of glyphosate, that's an extremely important job. So with multiple studies for each of the endpoints or hazard classes, like carcinogenicity, which we were looking at, where the classification criteria cannot be applied directly, i.e. to one single study or a group of similar studies, then all of the available information bearing on that determination of hazard is considered together. And this is very clearly stated in the regulation. We look at the quality and consistency of the data and give it appropriate weight. We look at both positive and negative results and assemble them together in a single weight of evidence determination. And this is important for the rest of my talk. Where the information from a single source alone is regarded as insufficient, the weight of evidence from several may lead you to a conclusion. And the role of epidemiology, so human studies, is specifically considered by the regulation and given a special place. About studies, there's a lot talked about studies, and Pavel mentioned, I think, the issues with glyphosate and steering scientific publications. In all of the chemical regulations in Europe, industry is responsible for generating the required data to demonstrate the safety of their products. That's their responsibility and has been for a long time. What counts is how reliable the study is and how outsiders can have confidence in that reliability. And key in this is the use of internationally standardized guidelines, usually promulgated by the OECD, for carrying out animal testing in particular. Added to that is the strength of good laboratory practice, which is an OECD-developed quality system. It's mandatory in the EU, EEA, the US, and Japan, and it's central to this credibility. If we have questions about the animal studies that have been carried out, then it comes down to the good laboratory practice check and the auditors who have looked at those studies. GLP-accredited laboratories undergo regular facility inspections, and more importantly, the archive study files are open to inspection by the National GLP Inspectorate. I think that's a very critical point when trying to decide for yourselves which studies are credible and which are not. Okay, on to glyphosate itself. First, talk about genotoxicity, so toxicity to the genome, mutagenicity in the regulation. A large number of in vitro studies, including mutation, chromosomal aberrations, using standard protocols were available, literally a huge amount. There was a broad set of both published and unpublished, largely good laboratory practice OECD studies, compliant, and in vivo reports mainly, as well as publicly available ones. We looked at 14 in total. So whereas a normal fresh dossier would have 1 or 2 of these, we're looking at 14 studies. It gives you an idea of the scale of the task here in sifting through all this material. There was also supporting combat assay. This is a different type of genotoxicity assay, both in vitro and in vivo. There were also studies involving human exposure to glyphosate-based herbicides. RAC concluded that glyphosate does not result in gene mutations or chromosomal aberrations. There was, however, some evidence of transient strand breaks to the genome. Our conclusion very clearly on the basis of a robust data set and a lot of data was no classification of glyphosate for germ cell mutagenicity is warranted. And that feeds into the next slides on carcinogenicity. The probability of something being a carcinogen is much stronger if it has a strong genotoxic basis. We found that it didn't. On carcinogenicity, we looked at animal studies, both rats and mice. Sorry to go into the gory details, but I think it's necessary. For the rats, we looked at a total of 9 studies, 7 fully considered. Oh, sorry, I beg your pardon. Go back one. There we go. I'm trying to coordinate 2 screens here because I can't actually see that one. Okay. For the rats, total of 9 studies, and believe me, 9 full carcinogenicity studies is a massive amount of data. 7 were fully considered. You'll see the reasons why below here. 6 GLP OECD guideline compliant. 5 were negative, but all were considered. Evidence for tumours in the pancreas, liver and thyroid seen in 1 study were considered in detail. 3 studies not compliant with GLP, etc. 2 negative, but 1 had positive findings and was considered. Evidence for pancreatic adenomas was considered in detail. RAC concluded the rat studies overall did not demonstrate convincing evidence of glyphosate-induced tumours. There's a low incidence but statistically significant trends for adenomas. Levels of carcinomas were in general not significantly increased and there's no progression to malignant forms observed. See if I can get it right this time. The mice. Total of 5 studies. These last for, I think, 18 months in mice, 2 years in rats. All studies were GLP OECD compliant. Evidence of renal tumours, hemangiosarcomas and malignant lymphoma present at low levels were considered in detail. Malignant lymphomas increased in 4 studies in 1 strain of mice, common tumour in Swiss mice and high control values in female CD1 mice. You will understand from this that in a lot of these toxicity studies, tumours occur both in the treated groups but also in the controls. Not all of them, but in quite a few. Renal tumours occurred at low incidences in 3 of the studies, 2 studies at very high doses beyond the limit that animals are usually subjected to. And these tumours are rare in CD1 mice, so this is some cause for concern. Hemangiosarcomas increased in 2 studies only at the highest dose. RAC considered all of the following: statistical significant, both pairwise and trends, dose response, which is particularly important, the biological relevance and consistency, etc., difference between the sexes and the high-dosed animals. Looking very briefly at the human data we studied, I think one of the key studies in the whole dossier is an American study from the American Health Society, a cohort study. It's prospective, so it's looking at a group of about 50,000 farmers in the Midwest of the US. They're followed over a very long period of time. This started in the late 1990s, as far as we know, is still ongoing. It showed no increased risk of cancer from glyphosate. It would be key, I think, to follow that study and to find out how it has developed since the last time it reported. For me, that is the key study which could change the paradigm here. The case control studies are retrospective, so they're carried out from cases in hospitals. They tend to be rather difficult because the sample can be quite small, and mainly the patients are asked from memory or asked by proxy or by questionnaires to fill in when they think they were exposed to glyphosate. It doesn't lead to a very strong base. However, there have been some weak associations with glyphosate published from some of these studies, and it's the special interest here is in non-Hodgkin's lymphoma. We found in RAC that chance, reporting bias and confounding factors could not be ruled out with reasonable confidence, particularly for the case control studies, and a causal relationship could not really be confirmed. So we concluded that the evidence from the EPI studies was insufficient to demonstrate carcinogenicity in humans. I don't think that's particularly different from IARC's findings, but I'm sure Kate will correct me on that if I'm wrong. Carcinogenicity conclusions in general. Animal studies were of sufficient reliability and relevance to allow a robust evaluation following the requirements of the EU regulation on CLP. In rats, convincing evidence of glyphosate-induced tumours was not demonstrated. In mice, while effects were observed, the incidences of findings were generally low. They weren't supported by findings at lower exposure levels, were generally seen without a clear dose-response relationships, which we would expect, and there was no evidence of progression to malignancy. The evidence from the EPI studies was considered insufficient to demonstrate carcinogenicity in humans in its own right. And finally, taking all of that delivered at high speed, taking a weight of evidence approach, no classification for carcinogenicity is warranted for glyphosate according to the CLP criteria. Thank you.
Very good. Thank you. And before Catherine will speak about the same topic and answer maybe your remarks, I will give the floor to two other speakers. And first is now Sylvia Lacorte from Institute of Environmental Assessment and Water Research. The floor is yours.
Thank you. Well, I'm Sylvia Lacorte. I am professor in environmental chemistry from the Spanish Research Council. And I would like to thank EuCheMS for the invitation to come here. So my talk will be based on the exposure of glyphosate, considering the use of glyphosate as a herbicide and how it finally ends up in humans. So as we all know at this stage, glyphosate is used in agriculture. It means that some of the agricultural products can have residues, and this can be uptaken by humans through the food or through water. I took data from Spain on the consumption of pesticides. While in Europe the patterns are probably different among countries, but that illustrates the magnitude of the use of glyphosate in Spain. So basically there are 350 pesticides registered, and in the, I don't know if it doesn't go. Anyway, in the pie cake we see the top 10 pesticides used in agriculture, and glyphosate accounts for more than 20% of the total. And when we look at the amounts, in Spain it is used in a total consumption of about 3,000 tons. And these 3,000 tons are sprayed in 3 million hectares, which corresponds to the surface of Belgium, as we are here. And then we look at the main crops where glyphosate is used in Spain again. So olives accounts for the first one, and then barley, wheat, and others. So basically, now what we are going to focus is the glyphosate in these crops. Well, this is glyphosate. By now we know the structure. It has more than 130 formulations, but there might be even more. The active ingredient is very different from the formulations. It can be from 0.7%, which is a very small amount, and this is used basically on gardening, to 68%, which is used in agriculture. It is applied in 60 types of crops, or maybe more, it depends on the country. And the amount, it is from 0.5 to 1.5 kilo per hectare, as has been said before. And it is used 1 or 2 times a year. It is a systemic pesticide, which it means that it goes into the plant. So this is why glyphosate could become a source of this compound to humans. The ECHA has classified it as a high production chemical, and there is a legislation which on the use which expires at the end of this year. So now we go into detail to physicochemical properties, which are very helpful to explain how this compound behaves in the environment. So basically, it is a very soluble compound. What means it's very soluble? It means that it will have a high affinity for water. It is non-volatile, so it will very rarely go into air. So it means that air will not be an exposure route. It has a high absorption to soil, and this high absorption, it is because of the chemical structure. Because of this high absorption, it has a very low leaching potential, so it will most probably not be found in groundwater. The degradation time in soil and in water goes from 15 to 100 and something days. So it means that after application, it can remain for a period of time. And it has a very low bioconcentration potential. So it means that in organisms, it will not or very rarely accumulate. So it must be stressed in here that this compound is very difficult to analyze. And when it means very difficult, it is very polar, as we have said, and it is very difficult to extract it from the samples that we are going to measure. So we have extraction difficulties, whatever the matrix. We have sensitivity problems to detect it at the nanogram liter or low microgram liter concentration, which is what is found in the environment. The methods are highly time-consuming with very complex instrumentation, and that makes that the analysis or the monitoring of glyphosate it has a very high cost. What is the result of this is that there are not very many studies done at the field level. And then there is another problem, which is the reliability of the results. Because of the method is difficult, sometimes it's difficult to compare the results obtained by different laboratories. So on the top, there's a chromatogram for who are interested that shows the traces of glyphosate and its main degradation product, the AMPA. So now we go to the field, because the starting point is the field, whatever field. This is a rice field. It could be any field. Well, in here, the fate or what happens with the glyphosate depends on the formulation, it depends on the dose, and it depends on the application method. So this is glyphosate in the field. Quickly, it degrades to AMPA. So we said it's not volatile, so it will very rarely go to the air. It will not leach, it will not bioaccumulate, but what happens is that it will remain in soil and accumulate in crops. And a part will run off, especially after application, to surface waters. So in reality, soils, crops, and waters could be the main uptake or source of intake to humans. So this is a wide-scale study that was done in Europe. If we look at waters, so waters, we have surface water and groundwater. We start with groundwater. There is evidence that theoretically it will not leach, and in practice it means that it does not leach. Out of around 3,000 samples monitored, only 1% had levels or residues of glyphosate, and the concentration levels in micrograms liter were very low. But when we go to surface water, which is on the top, we see that glyphosate and its main degradation products are found in around 50% of the samples, and the concentrations can reach levels of 400 micrograms per liter. 400 micrograms per liter probably does not say anything to anybody, but we have to consider that in Europe the reference value is 0.1 microgram per liter. So it means that the concentrations in environmental samples can be very high. If these waters are used for human consumption, then that could be a risk for the population. If we go to food, I mentioned here some studies that have been found in the bibliography, but in reality there are very few studies. The main residues have been found in crops, basically in wheat, soya, barley, even in bread, in farm animals, in fruit and vegetables. The real life is that there are very few studies, and it's very difficult to say if food could be a source of glyphosate to humans. And then in processed food as a result of the path through the food chain. And finally, also glyphosate has been detected in tap water, and the highest levels have been reported in the United States. Here we see, if we take the food, we see the maximum residue levels, which are settled by Europe in the regulation which is indicated above. This is the maximum levels that a food commodity can have of glyphosate. For instance, citrus, it can have 0.1 milligram per kilo, and mushrooms can have 50 milligram per liter. These are the two limits. So considering these levels, which are the maximum that could be found in food, we look at the exposure. So the exposure, it is measured. So at the end, we want to measure what is the magnitude of the problem. We measure the daily intake, and these units are given in milligrams per kilo per body weight per day. So if we measure the estimated daily intake, this is what we eat every day and the concentration of glyphosate in the food commodity that we are eating in our specific diets. And it's the sum of one day diet. So in the European population, this has been set to, considering these maximum levels, to 0.01 milligram per kilo. The acceptable daily intakes also suggested by Europe, they are 0.3 milligram per body weight in Europe. The EFSA suggested 0.5, and in the United States is 175. So if we consider the daily intake, it means that through a normal diet, we would ingest 3% of the acceptable daily intake, which is low. So it means that considering these limits, the exposure would be low. But here we all know how complex is the body, and we all know that the risks are different from different people. So in the upper side, it is mentioned the toxicity of glyphosate and its degradation products, which it is 2,000, so 2 grams per kilo. 2 grams per kilo, it is like a sugar spoonful of glyphosate. So the levels are very high. And for AMPA, it means 8 grams. 8 grams is a huge quantity. But there are other problems. So we look at the exposure and we look at the effects. And the effects can be various. And in here, according to the bibliography, it is mammary tumors, endocrine effects, some people say no myeloma, lymphoma, some people say. It has been detected in urine, but not in breast milk, and alterations in kidney. So there are other effects which is not, we could say they are more subtle effects. But concluding is that we have a big amount of environmental contaminants, prioritized contaminants are only a very few of what is used or what is present in the environment. So glyphosate, it is detected, we know it is detected especially in water. We have a lack of information in food. It's produced in a large quantity. It means that we have to have an eye on it, because specifically because of these huge amounts produced. Sorry, I cannot see very well. Well, it has effects, and why it is not legislated. So this is the question mark. And the needs that I see from a research point of view is that there's an analytical problem. The analytical problem is related to the difficulty to analyze this compound and to the costs related to this analysis. We have an environmental lack of data, especially for food, but also for water, and specifically on drinking water. And we have a toxicity lack of data, because sometimes we look at the burden or burden toxicity endpoints, but there are more subtle endpoints that could that glyphosate could have an effect at low concentrations. So I always like to finish with a picture of a clean environment and the need to preserve it. Thank you.
Very good, thank you, and thank you for sticking on your time. And now an enzymologist perspective on glyphosate from John Coggins, University of Glasgow. John, floor is yours.
Get the mic.
Sorry. Get the mic. All right. I'd like to thank the organisers for inviting me to participate in this discussion, which is a very important topic. I'm going to try and give a bit of a historical perspective, because I was somebody who was involved in glyphosate research really a very long time ago, in the 1980s when the compound was first used. And I happened to be the enzymologist who had the enzymes that Monsanto at that time didn't have, and I was able to do quite a lot of testing. I need, I think, in fairness to all of you, to declare my interest. I mean, I was heavily funded by ICI Agrochemicals throughout the 1980s. That's a company that's now been assimilated into Syngenta. Although I have given seminars both in DuPont and Monsanto, I've never received anything from them, so they've not supported my research, and sometimes my results have contradicted some of theirs. It's sort of fair to say that. More recently, I've been very active in the field of developing novel antimicrobial agents, and that research has been funded to a significant degree by AstraZeneca and BBSRC and by GlaxoSmithKline. So I think it's fair that those interested declared at the beginning. If I advance the slides. The compound glyphosate was first isolated or first determined to be herbicidal by John France at Monsanto. And it was a compound that had been investigated, and this is relevant to a bit of discussion earlier, as a water softening agent. So it's a very potent chelator, and that's relevant. Somebody was talking about cytochrome P450 inhibition. I mean, most metal chelators inhibit cytochrome P450s indeed. It was in many ways a dream discovery. In fact, John France won some very distinguished international awards for contributions to technology for the realizing it was a herbicide and exploiting it really rather quickly. It was shown quite early by rather crude analytical chemistry that it blocked the shikimate pathway, because compounds like shikimic acid accumulated in the poisoned plants on quite an epic scale. But at that time, our knowledge, particularly of plant biochemistry, was pretty weak. No one had ever purified any enzymes of the shikimate pathway from plants. I'm just going to flash up a couple of slides. I'm sorry the screens are so small. I'm used to presenting where there are large screens. But all you need to get from this apparently complicated slide is the shikimic acid pathway is a pathway by which plants and microorganisms make aromatic compounds. And they start with two compounds from central metabolism, erythrose phosphate and phosphenolpyruvate. And these are converted into a cyclic compound and eventually to a compound called charismic acid. And charismic acid is the common precursor of many vital things for plants and microorganisms, particularly the 3 amino acids, tyrosine, tryptophan, and phenylalanine, but also for very important compounds like ubiquinone and plastoquinone. And in plants, it's particularly relevant to note that the compounds that are important in the structure of plants, the lignins, are made from this pathway. And in many plants, as much as 30 to 35% of the carbon going through the plant ends up in lignin, for example. So inhibiting this pathway is very bad news indeed to plants. But it's also, and I'll come back to this in a minute, quite important to recognize that these pathways are also important, but for subtly different reasons, in all microorganisms. Bacteria and fungi have the shikimate pathway, and glyphosate has quite potent antimicrobial properties. In fact, in the 1990s, my lab, in collaboration with some American labs, demonstrated that it even had antiparasitic activity. And I'm ashamed to say that we didn't patent that, and Monsanto did patent it, although they haven't managed to make any money out of that. Anyway, one of the things that was helpful to research at the time, and that's the reason why ICI Agrochemicals invested money in my lab, is we actually had samples of virtually all the enzymes of the shikimate pathway, and in particular we had pure EPSP synthase. And a German lab, Nick Amrein's lab, and the worker was Hans Steinrücken, who works for Syngenta these days, they reported that the site of action was the sixth enzyme of the shikimate pathway, this EPSP synthase. And their work was done actually with fairly crude extracts of bacteria. That was the work, because the plant enzymes, as I've mentioned already, were not available. We, at that time, were working on the fungal EPSP synthase, and we had a significant quantity of it, and my graduate students, and particularly Martin Buchok, did some very detailed kinetic studies, which I'm not going to share with you in detail, but they were really very revealing. And I mean, subsequently, a year or two later, we repeated the work in the plant enzyme, which we were the first to isolate and purify. And the key findings here were that the compound glyphosate formed what is called a dead-end complex with the enzyme and the other substrate, shikimate phosphate. So if I show you a diagram, sorry, wrong way. That one, okay. In that diagram, you'll see the flow of catalysis goes through the middle of the diagram. So the enzyme forms a complex with shikimate phosphate. It's an ordered mechanism. And then it can either bind phosphenol pyruvate if it's going to its correct product and going straight through to product, or it can be sidetracked. And it's this sidetracking to the thing at the top of the screen there, an enzyme shikimate phosphate glyphosate complex. And that complex is very stable because glyphosate really binds quite tightly to the enzyme shikimate phosphate. And the only way the plant could escape the inhibition is if it had a very abundant local concentration of phosphenol pyruvate. Now, phosphenol pyruvate is a vital intermediary metabolite, and it doesn't occur in high concentrations. It's a valuable commodity in plants and other organisms, and it never accumulates in large amounts. So once the plant has been poisoned, it can't escape that inhibition. In fact, enzymologists have talked about this as an ideal kind of way to design an enzyme inhibitor that might potentially be a drug or a herbicide. So credit, in a sense, to John France and his collaborators in Monsanto. They found a wonder compound. In fact, one or two agricultural scientists have likened this discovery to the discovery of antibiotics, saying this was sort of a wonder compound. But as we'll see in a minute, there are some disadvantages to it, and I'll try and put it into some sort of perspective. But the compound, we've looked at the structure already, it's charged in solution. The diagram there is misleading. The phosphonate group and the carboxylate group will be ionized, and at most pHs there'll be a proton on the amino group. So it's a very charged molecule. That's part of the difficulty that was referred to earlier in analytical chemistry, simple water-soluble charged molecules are very difficult to analyze. And that's a headache. But there's nothing intrinsically nasty about that molecule. It's an amino acid derivative. It's a derivative of glycine. And it's not manifestly toxic. Somebody mentioned, what was it, one of the nerve poisons earlier. It's not at all like that. It's not a potentially dangerous compound, although one always has to worry about what metabolites might arise from these compounds, because some of those could become more toxic. I'm not going to dwell on this slide, but I want to tell you an anecdote that relates to it. When ICI was supporting my research, they were desperate to see if they could make an analog that would allow them to break the patent that Monsanto had. And we changed, with the help of the ICI chemists, we changed every single atom in glyphosate. I mean, that slide is incomplete. It just happens to be one I had from Martin Buchholz's PhD thesis. But it really is an isolated toxophore. In any way you tinker with that molecule, you lose the biological activity. There's one line there about glyphosate suggesting it's active, and I can tell you a slightly funny story at the expense of Monsanto. When I showed a slide similar to that when I was giving a presentation in St. Louis, John France sort of interrupted my presentation and said, I have to apologize to you because the sample which I think you got from ICI of glyphosate was contaminated with glyphosate, which accounts for the slight effect. But the isolated toxophore aspect of it has meant that it was very easy for Monsanto to defend that area of intellectual property, and that's why they were able to gobble such a share of the market. Now, if we look at the period after glyphosate really became successful in the sort of mid-1980s, lots of companies around the world in the agrochemical area thought, well, if inhibiting the pathway to an essential metabolite like an amino acid is a useful thing to do in plants, we'll try to do this in other pathways. And huge sums of money were invested in plant and microbial biochemistry then. But this hard work didn't yield any good competitor for glyphosate. And, well, those of us who were in competition, if you like, were looking for better compounds. Monsanto started to take advantage of the huge advances in plant molecular biology that occurred in the late 1980s and early 1990s. And they were able fairly readily to produce these Roundup-ready crops. These were crops into which they had inserted a gene encoding an enzyme, a form of EPSP synthase, which was resistant to glyphosate. Now, this is important. These enzymes were produced from microorganisms that had been deliberately exposed to massive doses of glyphosate. There were a series of publications, and I think the first commercialized gene in a Roundup-ready plant was actually isolated from the organism isolated from the sewage from their manufacturing plant. I think that's a correct story. And this emphasizes that, in fact, glyphosate in the environment can actually have an interesting effect on microorganisms. It can change their properties. Am I running short of time?
2 minutes.
2 minutes. Okay, thank you. So I'm going to show you two brief slides now. One listing some advantages and disadvantages of glyphosate, and then some conclusions. It does have low mammalian toxicity. We've talked, or at least some mentions already been made of possible dangers at quite low concentrations, but the crude sort of experiments that were done to test the product in mice and rats don't suggest that it's got any significant toxicity. It's very readily taken up and transported around plants. It's degraded into what I would regard largely as fairly harmless products in soil microorganisms. The main product isn't always aminomethylphosphonate, actually. It's more commonly sarcosine, which is N-methylglycosine plus phosphate. But it depends on exactly the population of microorganisms in the soil. It is amazingly effective at very low application rates. And I think this is something that in my final conclusion I'm going to point out, that in fact the excessive use of it is unnecessary. I mean, it doesn't need to be, applying it 5 to 10 grams a hectare can be very useful, not in 500 grams per hectare, and I think that's an important thing. It does facilitate this new approach to agriculture called low tillage cultivation, where year after year the same fields are planted with the same crop, and the previous year's crop are killed off with glyphosate, and then the resistant crop, soybeans produced like this, are planted there. There are weeds that have become resistant to glyphosate. The soil organism populations are perturbed by heavy use of glyphosate. And one issue that many have against Monsanto has been that the seeds for the resistant crop plants cannot be saved by the farmers. They're essentially F1 hybrid type things, and they must be purchased from the company, and that's how the company have extended their monopoly. Now, all right, we go to my last slide. Serendipitous discovery, it is an ideal enzyme, not toxic. But there are some disadvantages, and some of these are the disadvantages we see in medicine with antibiotics, for example. With antibiotics, you must not abuse them by overusing them. And I think in the herbicide field, I think we've got to think whether there need to be restrictions or regulations about this. And somebody's slightly referred to this earlier. One trick that farmers have used in recent years is applying glyphosate to kill off crop plants at the end of their growth cycle. In some cases, this enhances the yield, for example, of sugar in sugarcane. In other cases, it makes it cheaper to deal with the wheat. The wheat is killed off at the end, and it's easier to dry, and therefore the farmer can make more money, if you like, from his crop. I think that's undesirable because it leaves significant glyphosate residues on the actual plant products. In the case of the wheat, the flour will obviously be contaminated. And I think the other issue, this fact that the agrochemical companies own the seeds, there is, of course, a way around that. And in medicine, we've tried to move in that direction. Nowadays, in the third world, many countries can get cheaply important drugs against AIDS, for example, or vaccines. And these are deliberately subsidized, and organizations like the Gates Foundation promote that. It could well be possible that one could have a similar solution with these seeds so that they could be made more widely available. I'd like to stop there. Thank you.
Very good, Professor. Thanks a lot. And our last speaker in this panel is Catherine Gutton from IARC. And floor is yours.
Thank you, and I want to thank the organizers for the opportunity to be here, and I want to thank all of you all for your attention. I'm Kate Guyton. I'm from the International Agency for Research on Cancer in Lyon, France. And I'd just like to begin by saying I don't have any conflicts to declare with respect to any kind of financial interest related to the subject matter of my presentation. So, IARC classified glyphosate as a probable human carcinogen, that is group 2A, in March of 2015. This refers to the strength of scientific evidence that glyphosate causes cancer. The IARC evaluation was done according to our usual practices. All of the data, according to our process, has to be in the public domain and available for independent scientific review. And this is really essential for transparency. Additionally, the reviews are done by the world's leading experts without vested interests, and this is essential for independence. Now, the IARC does not make any recommendations in terms of policy actions following our monograph publications. At times, there have been risk assessments to understand the extent of potential cancer risk. Additionally, there can be measures to reduce exposures, even while additional studies are ongoing to better understand the risk. So what I'd like to do, I'm very afraid of the hammer, but what I'd like to do in the next 15 minutes or so is explain to you how we do our evaluations and really get into some of the data on glyphosate and hopefully answer some of the questions you may have. So you may wonder, if you had a big pile of studies, how you would turn it into one of these orange-covered books in which we publish our monographs. And the answers to that are given in the preamble to the IARC Monographs program. And this is available online. You can access it right now. It gives everything from procedural guidelines for how we select the participants who do the evaluations, how we manage conflict of interest, engage with stakeholders, and conduct the meetings. It also gives the criteria by which we evaluate the different lines of evidence and the decision process for the overall evaluations. And I'll go into that in the next couple of slides. So the first question you might have is, who does the evaluation? This is done by working groups that we convene. These are independent scientists without conflicts of interest. And this is who reviews the science. They do all of the writing of the drafts, and they are the ones who develop the evaluations. Now, there are other participants in the meeting. There is the IARC Secretariat. That would be me. So I coordinated aspects of the evaluation, but I didn't participate in that evaluation or do any of the drafting. We also have participants who attend the meeting but don't either write text or contribute to the evaluation, and these fall into three categories. We have invited specialists, and these are scientists who have relevant knowledge but also have a competing interest. We have representatives of governments and health agencies, and we have observers typically that come from the industry. They are there to observe the meeting but not to interfere in any way. So let's talk about the evidence that's considered, and it's really three lines of evidence. There are studies on cancer in humans, cancer in animals, and also on mechanisms. And these are brought together in an overall evaluation that I'll explain next. I do want to mention we have a chapter on exposure data. This is some of the issues that's been already quite eloquently described about how people may be exposed, how it's produced, how it's used, how it's regulated, and this kind of thing. But that isn't what weighs into our evaluation. Now, the evaluation process is what we call structured expert judgment. It's a two-step process. In the first step, the evidence in each of these different lines is categorized according to the definitions that are in our preamble. So for the evidence of cancer in humans and cancer in experimental animals, the possibilities are sufficient, limited, inadequate, or evidence suggesting lack of carcinogenicity. For the mechanistic data, it's really a different question. It's whether it's strong data, moderate, weak, and also whether those data are coming from exposed humans. This type of data is very important to a working group. If it's a biomarker of a cancer that's, for example, in exposed humans, highly exposed workers, things like genotoxicity in populations that have been exposed, this is always going to weigh heavily on the minds of a working group. Now, the second step of the process, these are brought together in a very formal way. This is described in the preamble once these initial conclusions are reached. Now, having done 1,000 evaluations, these are how the results have come out. And you can see most of the results, most of the compounds have been evaluated into group 3, not classifiable. That's more than half of the evaluations that we've done. There's only one lonely compound down in group 4, which is probably not carcinogenic to humans. And that's really because, according to our preamble, we evaluate substances that have a suspicion of carcinogenicity. So perhaps it's not that surprising. Now, fewer agents are into the higher categories of Group 1, Group 2A, and Group 2B. Group 1 agents are principally those where there is sufficient evidence of cancer in humans. This is things like tobacco smoking, asbestos, benzene. These are the established human carcinogens. The Group 2B agents are largely those that have tested positive in animal bioassays, usually 2 independent animal bioassays according to our judgments to reach that sufficient level of evidence. And Group 2A is largely compounds for which there's some evidence in humans and sufficient evidence in experimental animals. But there are agents there because of mechanistic considerations as well. So let's talk about how this all comes together in space and time. The meeting gets announced by the IARC. We publish this on our website. We describe the topics. We indicate what conflict of interest we are asking people to declare, and so forth, and we give the schedule when we are asking for data, when we're asking for experts. We convene the working group, and they are doing their study evaluations and writing their initial text during a period leading up to the actual meeting. Now, before the meeting happens, about 2 months in advance, we publish who will be at the meeting, who will be the participants, and what interests those people have declared. And as I mentioned, there's these different 5 categories of participants, and all of that is evident, so you know who is going to be at the physical meeting, which happens in Lyon. And during this meeting, this is really where the working group debates the science and reaches their conclusions. Following this, we publish a summary together with the working group in the Lancet Oncology a couple of weeks after the meeting, and sometime later we will publish the fully referenced monograph. So let's see how this came together for the glyphosate monograph. It was about a year-long process by which the working group was working on their drafts. I have all the dates there in terms of when we published the announcements. Now, because of the intense interest in the monograph, we did work very quickly after our evaluation to share our references with health agencies and publish our monograph. Now, in terms of participants in the meeting, we did have observers from Monsanto, from the European Crop Protection Agency, from universities. We also had representatives from the U.S. government and other governments, and we also had our working group, which did not have conflicts of interest. Now, our meetings are open to these observers, and we were very pleased that they are there. However, it is the working group who took the evaluation. As I mentioned before, it's a transparent process by which everyone at the meeting has access to the data and they have access to the drafts. And finally, we did publish that monograph, and it's still on our website, and you can go get it right now and start reading it if you like. And while you're doing that, I will tell you a little bit more what's inside. So this is really a systematic review of the literature evidence. If you go to PubMed right now and type in glyphosate, you're going to see you get a lot of hits. And we pulled together about 1,000 studies that were initially screened, focusing in on studies of glyphosate in laboratory-type studies. These were of glyphosate itself as well as in its formulation. Studies of cancer in mice and rats, as we've heard, and particularly studies of DNA damage, but also other effects related to carcinogenicity. There were also human studies. These are people who were exposed in their jobs, primarily, or in their communities, and studies with endpoints such as DNA damage, but also of cancer. Altogether, the published monograph cites about 250 of these references. So let's talk next about the evidence of cancer in humans. Now, we've talked a lot about agricultural uses of glyphosate, but another heavier use, I believe, is in forestry applications. So these forestry workers are actually quite highly exposed. And there were 2 types of studies that were available to the working group. The first is a case control study. This is where you have cases that occur, and you go back and you find controls and do a comparison. Now, these studies came from 3 regions in the world, in Canada, in Sweden, and the United States. The advantages of a case control study is you can get a lot of cases because you know what you're looking for and you're going after it. In these studies, there was an increased risk of non-Hodgkin lymphoma that it could not be explained by exposure to other pesticides. Now, there's also a cohort study, which is a very strong design. This is where you take a cohort of people and you observe them over time. Now, when you're interested in cancer, it takes a very long time for these events to occur. This study is still relatively young at this point in time. It's being done in 2 states in the United States. It doesn't have so many NHL cases at present. And so far, there was no significant increase in risk reported from that study. Now, there's another type of analysis that this particular working group undertook, and many working groups do this, is to do a meta-analysis. This includes all the data from all the studies, and you bring them all together, and you try to understand what is the risk if you looked at all this data together. And when this was done, there was an increased risk with the meta-risk ratio of 1.3. And I just want to point out that I-squared, which is an indication of heterogeneity. So if your data is all over the place, your I-squared is very high. This is not the case here. So this is a relatively consistent body of evidence. And because of that, the working group concluded that there was limited evidence for cancer in humans. So let's now talk about the animal bioassay data. We had a lot of discussion about this at the meeting because the working group, because of our rules, had to use data that they could really independently judge and not accept the judgments of the manufacturer or anybody else. So they had to have enough information to really judge us independently. And when they did that, they really only had 2 studies in mice. Both of these were positive. And one of them is we've heard earlier, there was these rare cancers. Rare cancers can be extremely important in identifying a hazard. It's a very rare event, and it probably wouldn't happen if that agent wasn't there. But there's different challenges, which is that you're not going to get very many events. You can use statistics to rule out chance, which is what we do according to our procedures. These were highly significant results. You had tumors in the absence of toxicity, and we did our evaluation fully in line with our procedures, which are the accepted principles. And based on this, a causal relationship was established from the animal database, and that led the working group to conclude sufficient evidence of cancer in animals. Now I'm going to talk briefly about the studies of genotoxicity. We know that genotoxicity can lead to cancer, as we've heard earlier. I think people accept that. There were not any studies of, let's say, manufacturing workers or other types of workers, but there were some studies of people who had been sprayed in their communities. Not very many of them. And in these studies, what occurred was when there was a look at their blood, before and after exposure, in the same individuals, you had an increase in DNA and chromosome damage. Chromosome damage in the blood is one of the best predictors of cancer outcomes in humans. So those are studies of the glyphosate formulations. There were other studies of glyphosate formulations in experimental systems, including human cells and animal cells. And in there, we also did see this chromosome damage and DNA damage. And similarly, that's the case also when glyphosate itself was studied. I do want to point out that glyphosate was negative for mutation in bacteria. This can be very challenging because the EuCheMS assay is actually pretty reliable and will predict the outcome of animal bioassays in many cases. It's a very, very good predictive value. So being negative in that initial screen, it can be difficult, but of course bacteria don't have chromosomes, so if you're interested in chromosome damage, you have to go to other test systems, including these studies that I've mentioned here. So let me now talk about these 3 different lines of evidence and the conclusions. So for the cancer in humans, for non-Hodgkin lymphoma, there was a conclusion of limited evidence. These are studies of people who were exposed in their jobs. They were exposed to glyphosate formulations. However, they were formulations used at different regions and at different times. And therefore, that led the working group to really say it was glyphosate as the causative agent with limited evidence. The study of cancer in animals, as I mentioned, these are studies of pure compound. There was an observation of these rare cancers and therefore sufficient evidence. And finally, DNA damage. I didn't talk about oxidative stress, which is another important factor. Strong evidence. These few studies of real-world exposures with people being exposed in their communities through spraying operations, but also a fairly robust data set of experimental studies of both glyphosate and its formulations. And altogether, this led the working group to classify glyphosate as group 2A, probably carcinogenic to humans. So hopefully I have one more minute. And I just wanted to point out, we've often been asked, well, why did you evaluate glyphosate and how did you decide to do this among all the pesticides that might be of concern? We had many, many pesticides nominated for evaluation. We determined that manual selection could be prone to bias, so we took a really systematic approach to this. And we published this, and you can look it up. But briefly to say that the size of these dots here indicate the amount of evidence that we found to be available from public databases. How close together indicates their chemical similarity. You can see our friend glyphosate there on the top panel. And just because there's been lots of studies doesn't necessarily mean that there will be a higher or different IARC classification, but it does mean that there's interest, and that is one of the main motivations. And on my last slide, I just also would like to put into perspective, similar to all the agents we've evaluated, the pesticides, most of the ones we've looked at, we've placed into group 3. Some of these more recent ones, based on this approach that I just showed you, we were able to identify those with a substantial body of evidence, and these were classified in group 1, 2A, or 2B following those. So I'll stop there, and thank you very much for your attention.
Thank you as well, and you just closed our first panel. Before I give the floor to the coffee, I want to announce then we will follow in the next panel with Jack Heinemann from University of Canterbury, New Zealand, on antibiotic resistance in human pathogens and influence of glyphosate, and we will continue with Thor Midtvedt from Karolinska Institute on ecotoxicological consequences of glyphosate, Peter Klausing from Penn, Germany on weight of evidence approach, Helmut Burzer-Schaden from Global 2000 on glyphosate and cancer, and Hartmut Frank, University of Bayreuth, on toxicological and environmental chemistry consequences of glyphosate. So stay with us and enjoy in next 5 minutes your coffee, and we will continue at 4:05 exactly. My friends, I'm afraid that we have quite a busy program for next panel, so I would like to ask you to take your coffee to your seats and as a first speaker, send us a video message. We can start with this video message, and you can easily drink your coffee hearing this video message. So I would like to ask and encourage everybody, take your seat, please, just now. Yeah, I know that it's always a problem to get people back to the seats, but please take your seats. Stop chatting. Yeah, please. Please. They don't hear me. They don't see me? Ah, now. Very good. Well. And we have a video message from Professor Jack Heinemann from University of Canterbury in New Zealand, and he spared our money for paying a flying ticket, and he sent us a video message on how herbicide can induce multiple antibiotic resistance in human pathogens, which probably will be very alarming. Let's start with that.
Kia ora. Thank you very much for having me at this important conference discussing the effects of herbicides, particularly glyphosate, on the many different ways in which they can influence human health and the environment. I hope my video is not too boring. It is my debut in the film industry, but I do appreciate the fact that you've saved me many, many hours on an airplane coming to Europe. I'm here to talk to you today about our work looking at the effects of herbicides, including herbicides such as Roundup, on bacteria, in particular those bacteria that have the potential to cause disease in humans or our domestic and farm animals. We live in an antibiotic-intensive world. Since World War II, with the introduction of mass-marketed antibiotics, we have produced millions of tons. And we've known for decades that our use and pattern of use of antibiotics was going to cause resistance among microbes. This picture of two different magazine covers illustrates that we've been aware of the problem for a long time. If you look very closely, you'll see the dates on these magazines is 1994, the year that I transferred from work at the U.S. National Institutes of Health to the University of Canterbury. So 23 years ago, we were already noticing the decline of the effectiveness of antibiotics. Yet little has been done to salvage these important medicinal compounds. Antibiotic resistance is estimated to cause a unique 700,000 deaths each year. This is projected to increase to 10 million by 2050, with a cumulative cost to the world economy of US $100 trillion. The loss of antibiotics as a foundation medicine will have more effect than just more deaths. Routine operations will be denied for the fear of an uncontrollable sepsis. And antibiotics underpin our agricultural productivity, particularly with livestock, but also with crops where antibiotics are routinely used to stop bacterial infections. In fact, more than half of all antibiotic production is used in agriculture. Importantly, less than 20% of the antibiotic given to an animal is metabolized, which means that some 80% of the antibiotic use in animals alone directly enters the environment, generally through feces, manure. Antibiotics are now so commonly used and at such scales that antibiotics can be detected in the atmosphere. And frighteningly, so can the genes that make antibiotics resistant, make bacteria resistant to antibiotics. Antibiotics are a form of a biocide. They're intended to be toxic to bacteria and not as toxic to us, to people or to animals. Other kinds of biocides are things such as pesticides, including herbicides. Now, in the United States, the Environmental Protection Agency permits over 200 different pesticides just for lawn care. This is an urban use. And these are often mixed together in complex chemical formulations. The U.S. Fish and Wildlife Service reported that home use uses 10 times the herbicide concentration that farmers use on crops. And indeed, our exposures are nearly ubiquitous. If you walk down the aisles of a hardware store, you're breathing in a cocktail of pesticides because they're often packaged in porous materials for safety reasons. Our work has focused on the 3 largest use herbicides in the world. They are those based on the active ingredient dicamba, the active ingredient glyphosate, and the active ingredient 2,4-D. In New Zealand, these are sold commonly as the herbicides called Camba, Roundup, and 2,4-D. Roundup is the heavyweight world champion of use, and in particular because it is tied to use with large monocultures of genetically modified crops. There is an overlap in the potential to be exposed to either a herbicide or an antibiotic. These overlaps are routine in our experience. For example, in the agricultural settings, livestock may be exposed to herbicides through spray drift. In countries such as New Zealand, there is no stand-down period between applying Roundup to a pasture and grazing dairy cows on that same pasture. So they can also be exposed through residues on their food. Honeybees, in particular in countries that use herbicides during the growing season, may be simultaneously exposed to the herbicides used in the crops and in their hives, where they are often prophylactically treated with antibiotics. Remember all those non-metabolized antibiotics that come out in manure? That manure is often used as fertilizer, so spread again onto fields where it is sprayed with herbicide. And even in urban settings, our exposures can be high. What I'm showing you here is a picture of Christchurch on our large central park called Hagley Park, where you see young women with their babies and prams walking through grass that is notified as having recently been sprayed with a weed killer. And we can have vectors between park use or rural use and our food, vectors such as blowflies, which can land on feces that have been treated by herbicides, fly into our windows, and land on our dinner. Our work has concentrated on the sublethal effects of herbicide exposures. Now, herbicides are biocides, and so are antibiotics. But herbicides, for the organisms that we work with, the potential disease-causing organisms, Escherichia coli and Salmonella enterica, they're not as potent as most commercially available antibiotics. Nevertheless, bacteria respond to them as toxins. What you see in this slide, in the upper left-hand corner, is a titer of the population of bacteria, in this case, Salmonella. The numbers that you see on that dish indicate the factor of 10 dilution. So we've diluted the culture 1,000 times, 10 to the 3, to 10 to the 9 times, or a billionfold. And you see a single colony under the number 9. That tells us that per milliliter of solution, the population of the bacteria was a billion per milliliter. If you look at the dish just below, where we've listed it as Camba plus no antibiotic, this is that same culture now put onto a medium that has the herbicide Camba incorporated into it. And again you see 10 to the 9 bacteria per mil. The colony is smaller, telling us that the bacteria are dividing much more slowly in the presence of the herbicide. But they are not being killed by the herbicide. We're recording just as many bacteria surviving on that medium with herbicide as on just normal media. If you look in the upper right-hand corner, this is the same culture of bacteria now exposed to medium that has an antibiotic in it. And you'll see there are no colonies at all. The antibiotic concentration we're using is lethal to all the bacteria in that population. However, if we put the bacteria onto a medium that has both the herbicide and the antibiotic, 100% survive. The herbicide has somehow and immediately protected the bacteria from the effects of the antibiotic. When they're exposed simultaneously to herbicide and antibiotic, we see 1 billion bacteria per milliliter, just as if there were neither herbicide nor antibiotic on that plate. We measured the effect of these 3 different kinds of herbicides on 5 different kinds of clinically useful antibiotic classes. And we measure what's called the MIC, which is the minimum inhibitory concentration of an antibiotic. The MIC is used by physicians to calculate the amount of antibiotic used to clear an infection. Changes in the MIC can therefore influence the efficacy of a therapy based on antibiotics. And what you see in our table is that sometimes a herbicide increased the susceptibility of the organism to the antibiotic, and sometimes it increased resistance to the antibiotic. On the rare occasion, it had no significant effect. The vast majority of exposures caused a change in the MIC. And all changes in the MIC may have important clinical or evolutionary consequences, whether it is to increase susceptibility or to directly increase resistance. The effect ranged from approximately a 2-fold change in the MIC to as much as a 6-fold change in the MIC. These are important numbers because as little as a 2-fold change in the MIC of some antibiotics can mean that the majority of patients receiving that antibiotic get too little to clear their infection, or to clear their infection in a time which reduces the potential for them to infect others with the same bacteria. These numbers, therefore, are clinically important. So what we found from our research is that the commercial herbicide formulations induce an antibiotic response in medically relevant bacteria. The effect is large enough, 2 to 6-fold change in the MIC, to theoretically significantly undermine therapy. The herbicide effects were detected at concentrations that are above currently allowed maximum residue limits of the herbicide in human food, but they were well below application rates recommended on the label that are used both in home gardens and in agricultural settings. The title of this conference is Glyphosate, Harmless Tool or Sneaky Poison. Our research says that there is a third thing that glyphosate can be. In our hands, glyphosate and some of the other herbicides appear to be vaccines, vaccinating bacteria against the effects of our medicine. Thank you very much for having me, and I'd also like you to simply acknowledge those who have done this research with me. Kia ora. Thank you.
Okay, finished. We cut because we are a bit late after on our schedule, so my assistant cut him, but it was only saying thanks, and I would like also to thank to Jake Heinemann because this sounds quite alarming, by my opinion. And now I'm very happy to give the floor to Tor Midtvedt from Karolinska Institute, and he will elaborate on glyphosate and its ecotoxicological consequences. Please, floor is yours.
Okay. I'm very glad for the invitation to come here, and I will try to go briefly. First, I will try to go through a little bit of the history you have heard before, and then I will concentrate upon some ecosystems that are attacked by glyphosate. As you have heard, it's an old compound. And when it started to be used, then it was just for in the beginning of the season. But later on, when they then created the glyphosate-resistant seed, then it could use all through the growth season. And nowadays, it can be used to end the growth season. That makes a completely different story upon the effect on the soil. So let me go. And if you're going, it's a lot of information to get out there. May 1st, then I checked in PubMed, it was 2,420, and today it's 2,430 publications. So it's constantly coming more and more publication out there. And also in the very first publication, it was shown that this drug could affect microbes. It was the early one, was cyanobacteria, and that's the same as green algae out there. And that is of importance in many ecological systems. And later on, just 2-3 years after, it was also that it could influence upon Escherichia coli, a very common. All of you have that microbes in your intestinal tract. It can cause infection, but it has indeed a physiological role to play there. Let me go further. And then the effect on the microbes is just the same as the effect on the plants. It stops production of some amino acids. We cannot produce these aromatic amino acids, so we have to get especially tryptophan from plant or from microbes. And out in the soil, then you have two kinds of microbes. You have those that produce tryptophan or those amino acids, and you have those that are depending on these microbes. And when you are then using glyphosate, you will simply kill the microbes that are producing tryptophan, and after a while that will influence upon the microbes that have to be given microphan. So that, from that point of view, you will always have a quantitative. You will not have a quantitative disturbance, but you will have a qualitative disturbance in the soil microbiology. That is complicated to investigate. If you go out in your garden and take up soil, in 1 gram of soil in your garden, you will have more than 4,000 different microbial species present. If you are then using glyphosate, you will kill maybe half of them, those that are producer, and after a while you will influence upon the rest that need to have. So from an ecological point of view, then the soil microbiome is of the most importance when we are discussing effect of glyphosate. For it change the base for all growing in that soil. And not just to mention one that will be over one of the slides, glyphosate is not only essential for the growth, but breakdown product of tryptophan, the main aromatic amino acids that are inhibited, that act as a plant hormone. It will then stimulate the plant to increase the number of root hair. so the plant will grow faster. If you have no tryptophan, you will have no production of that growth improver, so the plant will grow slower. Those areas, the effect of glyphosate on soil microbes are the least one investigated and should be far more investigated. Then I can go through some of the other one. I will go further on. And then you can think about what are known about other ecosystems in the soil. If you are taking earthworms, if you have a garden, you do like to have a lot of earthworms there. That's of importance. It has been some studies on the effect of glyphosate on earthworms, and they are reduced in activity. The mechanism by which the earthworms are not functioning as they are under discussion. It may be that it is effect on the earthworm themselves, but it also, and most probably, that it is an effect of the ecosystem in the gut of the worms. Earthworms, have an intestinal tract, just as we have. They are depending on the microbes to work there, and if the microbes are disturbed, then you will have less activity in your intestinal microbiota and less activity in the earthworm. We could take ecosystem after ecosystem, and then, if you can, but earthworms is very much disturbed. We can then go to other insects. That could be nice to get rid of insects, but they have to be there. They have some functions to, and it has been done a lot of studies on glyphosate on insects, and it's nearly always a reduction in number, and sometimes they are completely eradicated. If we take the bees, it has been, as you are aware of, you have a lot of problems with bees all around the world. And it's at several theories by which glyphosate influence upon the death of bees. Maybe the last one came up just some few weeks ago. And that was an investigation showing that on the pollen, in all plants have pollen, and on pollen you have a very specific microbiota. And that microbiota is taken up by the bees, who has really a very specific intestinal microbiota. If you then use glyphosate and other herbicide, not only that one, you will interfere drastically with the microbiota on the pollen, and secondary with the microbiota in the bees. So far, no studies have been done with the bees themselves and to follow that, but the possibility is indeed that. So you should take a look on that publication. It's Michael Zaslow, who is the author of that one, and he was in fact one of those guys that started to investigate defensins 25 years ago, and that was in frog. I go further on. What about amphibian? Can they be influenced upon by glyphosate? It has been done a lot of studies also on that. And some studies are saying no influence, other ones say it is an influence. If you are doing it very, very clearly, you will find the younger the amphibians are, the more consistent in the influence. That's the main message to take home from that picture. And it's a general statement on that slide, stating that when you are studying glyphosate on organism, you should follow it all through life. And in many of these investigations, the younger the individuals are, the more profound is the influence upon glyphosate. So that should be something to remember for the regulatory authorities. I'll go further. So you can, birds, they are being used as, you can say, as they are important if you want to follow alteration in ecosystems. And there are very few studies working with birds and toxicology of glyphosate. It is one study from Mexico working with ducks, but here in Europe it's very, very little done regarding the influence of glyphosate on birds. And we up in Scandinavia, we are a little bit afraid of that. For our birds are passing Europe twice a year, and especially in the autumn, if glyphosate is used to stop the growth of wheat and other plant, then the birds came and take the rest, so they are really they will have a heavy burden of glyphosate when they're going south. And that part of the ecosystem should be far more studied than it has been so far. And since we come from Scandinavia, what about fishes? And especially in Norway, I'm originally a Norwegian. In salt water, then there's very little glyphosate. But in sweet water, as we have heard, it can stay there. And it has a prolonged half-life in sweet water. It can be over 200 days that glyphosate can stay. The half-life of glyphosate can be over 200 days in sweet water. It depends on the pH and temperature and so on. But anyhow, as you remember, I stated that glyphosate were very toxic to cyanobacteria that are of importance as food for fish in the sweet water. So that also a part that should be far more studied than it has been done today. What about us? A little bit, man is also a part of ecosystem. But first, as you are aware of, as we have heard, it is now more and more weed that are becoming resistant to glyphosate. You can have a different strategy to attack that. You can increase the amount of glyphosate used. But anyhow, it may be the main thing for get glyphosate out of use. If all weeds became resistant, then it's no use to use glyphosate. So then maybe Mother Nature is doing the job instead of the regulatory and politicians in the future. I'm a little bit provocative, but I'm putting my money on Mother Nature. And also what we have heard about the microbes now. And microbes are becoming also resistant. My friend from New Zealand, he told about the different mechanism. And if the resistance in microbes are due to, are usually not due to that glyphosate is destructed, but it is due to that it is spitted up from the microbe, so it will reach the next and so on. So therefore, if and the resistance is on plasmid. And if it is coupled to human important microorganism, as it was indicated by my friends from New Zealand, then it will be of very great importance for human medicine. I'm running out of time, so I have to speed up. Yes, I am. So, we are exposed, humans are exposed to small amount of glyphosate sometimes during drinking water, sometimes during plant, and it's reached our intestinal ecosystem. And now it's more and more realized that many of these intestinal trees, they are in disbalance. And it has been discussed, what about autism spectrum disorder? It has been discussed in metabolic, it has been discussed in cancer and so on, that it can be also infect our cell and our health. So therefore, from my point of view, as an ecologist working in the field longer than any of you, it's very, very simple. They are going there. They are protecting themselves. But we are going to eat the food they are using glyphosate on. So from my point of view, it's just one conclusion to be giving. Thank you for listening.
Thank you, Thor, very much for this, ecological perspective, which is, I think, the angle we should use more often, mainly in decision making and legislative process.
Thank you for that words. I like them.
And the next speaker of ours is Peter Klausing, professional toxicologist, and he will elaborate on carcinogenicity of glyphosate and the weight of evidence approach. The floor is yours.
Thank you very much for the opportunity of presenting here. I'm a retired toxicologist, now volunteering with the Pesticide Action Network Germany. So you will have an NGO's perspective with a scientific background. That's what I would claim. I will return to the mouse carcinogenicity studies, and we will have a more detailed look at this. But I think to better understand the data I'm presenting, I will just refresh your mind with two pieces of information before looking at the data. And the first is the CLP legislation, the European legislation for classifying compounds as carcinogens or not. And I think you will keep in mind that currently glyphosate is classified as not carcinogenic. For a class 1B carcinogen, the abbreviated version of the legislation is that if you have two or more studies, independent studies, showing a significant increase in tumor incidence, this would be something like class 1B carcinogen. And if there is not sufficient evidence but limited evidence, you will look, according to this definition, at an increased incidence in a single experiment or unresolved questions concerning the interpretation of data of the studies, or benign neoplasms, benign tumors only. The other piece of information concerns statistics. There are different statistical methods looking into these significant increases of tumor incidences. And there has been a lot of discussion about the differences between IARC and the European authorities with regard to coming to a different conclusion. And the most frequently used argument was they have looked at different studies, or the EU has looked at more studies than the IARC. I claim the major difference is they have used different statistics. The EU uses so-called two-tailed tests. I will not go into the details what this means. What this means you will see, but what the background of this is. And they prefer using pairwise comparisons, whereas the IARC is applying trend tests and one-sided tests. This difference is between IARC and the EU is twofold. Number one, the EU approach from the statistical perspective is weak, and the one from the IARC is powerful. And the second difference is that there is guidance for assessing carcinogenicity studies from the OECD, which essentially recommends the approach of the IARC. So let's look at the data. In the beginning, and this was in March 2015, there was this approach by the BFR based on the studies submitted by industry, using a pairwise two-tailed test. And when you look at the 5 mouse carcinogenicity studies and look at 3 different tumor types, there is just one tumor which had a significant increase, malignant lymphoma, and all looked fine to claim that glyphosate does not present a carcinogenic hazard. Shortly thereafter, or essentially in July 2015, the IARC monograph was published, triggering a reanalysis of these mouse studies by the BFR, this time using trend tests, but still two-tailed test statistics. So I put the weak in parentheses, and something which also needs to be kept in mind that this OECD guidance 116 says that significance in either kind of test, either pairwise or trend test, should be considered as sufficient to declare significance. Looking at the data, the picture changed dramatically. Before we had just one test, and the pairwise comparison in the 2001 study, which showed significance. Now with trend tests still two-tailed, we have significant increases in all 5 mouse studies. We have increases in different tumor types repeatedly. And the question is, or the question I think was for the authorities, for the BFR, for the EFSA, how to deal with it. So the next step was looking into what is called high-dose effects. These are marked slight red, which decrease the significance using the weight of evidence approach, which decreased the significance of the findings because it was limited to doses up to 4,000 milligrams per kilogram. And one study which I think is and was and still is a crucial study is the study of 2001, which showed significant increases of tumor incidences in 2 studies. And when we will look further at the data, you will understand why it was essential to kill this study and take it out of consideration and weaken the consideration for the 1983 and 1997 study, and at the same time keep the study of 1993 in, which I cannot, for time reasons, cannot go into detail, is invalid with regard to malignant lymphoma. But if you disregard this invalidity, you can show that there is no significant increase in malignant lymphoma, and this will be the type of tumors we will continue to look at. Next, a quick look at why the suspected virus infection caused to exclude the study of 2001. When you read the conclusion of the EFSA, you can read that this study was not acceptable due to viral infections that influenced or could influence the survival as well as the tumor incidence. When you read the draft CLH report of 2016, you find what the scientific basis is for this exclusion. It is mentioning by a US EPA observer, and it was actually Jess Rowland who was participating in this telephone conference, and who mentioned that this study had been excluded by US EPA, and picking up on this without any further evidence, without any evidence in the study report and without any explanation last week by Jose Tarazona, with whom I had the pleasure to discuss for an entire day. There is no other evidence to exclude this study. So when we take, and that's why I think we should take the study of 2001 back in and should take the study of 1993 out. And in this case, we will see that we will have in the malignant lymphoma, using the one-tailed test, comparing pairwise and trend tests, we will have in 3 of the studies, we will have significant increases in the trend test and also significant increases in the pairwise comparisons in 2 tests, the one of 2001 and of 2009, and the study of 1993 is falling out as an argument to be proof for no carcinogenicity or no malignant lymphoma. This is particularly important when you look at the authorities' main arguments with regard to the so-called weight of evidence approach. The main arguments is lack of significance, or where, you can read it in a recent publication by Tarazona and co-authors from a couple of weeks ago, and you can also read it in the documents. Lack of significance in pairwise comparisons. We can see we have, when we accept the 2001 study, we have significant increases also in the pairwise comparison. The second argument is lack of consistency in multiple animal studies. You have 3 studies with significant increases for malignant lymphoma. You have 1 study of 1983 which has no significant increase, and you have an invalid study which I would prefer to exclude. You have the argument of that these effects are only seen at very high doses, and high doses, according to the OECD guidelines, are defined as a limit dose of 1,000 milligrams per kilogram. This limit dose can be argued and questioned, but we'll keep it for now and look at the studies, and we will have the study of 2009, which has a top dose of 810 milligrams per kilogram. We have the study of 2001, which has a top dose of 1,460 milligrams, but you should keep in mind that glyphosate is resorbed only by 20% of the administered dose approximately, and you have just one high dose study, one true high dose study, the one of 1997. So when you look back at the CLP regulation, having in mind that tumor increases in 2 or more studies would qualify for a 1B categorization, then you have, for glyphosate, 3 studies with a significant increase in the same tumor type within one sex. And this is another aspect, but this is not a prominent aspect in the weight of evidence approach. You have 2 studies with a clearly dose-dependent increase. When you look at the incidences below that, you have an increase from spontaneous tumors in the control group of 10 to 15 to 16 and 19, malignant lymphoma in the male group of the study of 2001. And you have an increase from 0 to 1, 2, and 5 in the study of 2009. To conclude, my question is, what was driving the decision for not even using category 2 classification for glyphosate by the European authorities? Thank you for your attention.
Yes, thank you. And it seems that our meeting gonna be really fruitful, because I'm convinced that we have to ask these questions during following process. Very well. Next is Helmut Burtsche-Schaden from Friends of the Earth, Austria, and he will elaborate a bit on glyphosate and cancer with subtitle Buying Science. The floor is yours.
Okay, thank you. Thanks for inviting me. My name is Helmut Burtsche. I'm a biochemist working for the Austrian organization Global 2000, and I'm one of 3 authors of the report Glyphosate and Cancer, Buying Science. Another author is Claire Robinson, sitting here, and Peter Klausing. And our report is about the quality, so now I hope it works, about the quality of science that has been either sponsored or performed or even ghostwritten by the glyphosate manufacturers, and with its influence on the cancer assessment performed by the German Federal Institute for Risk Assessment. BFR's assessment was eventually confirmed by EFSA and later by ECHA. Moreover, BFR's leading experts were also active in the first European Union cancer assessment. They chaired also the ECHO team that evaluated afterwards their first assessment. They chaired the JMPR meeting in 2004, and they chaired the JMPR meeting 2011, which could be seen as indicative for revolving doors between or within European institutions assessing cancer on glyphosate, and also to international organizations. And this is of importance because the German authority, and even more Monsanto, repeatedly emphasizes that scientific bodies all over the world consistently conclude that glyphosate is not carcinogenic. A possible explanation could be that they have all the same source industry. And this is at least the case with the European Union's cancer assessment, which, as we will show now, has relied almost exclusively on unpublished regulatory studies that have been conducted or commissioned, that have been sponsored and finally evaluated by the manufacturers. And they relied on published review articles that have also been commissioned and evaluated or sponsored by industry. Published academic literature independent from industry that under pesticide regulation 1107/2009 must be considered was dismissed as being either of limited evidence or of no evidence at all. Based on this evidence, BFR concluded that there is no evidence of carcinogenicity in humans, no evidence of carcinogenicity in animals, and no evidence of genotoxicity. In March 2015, these conclusions clashed with the assessment of IARC, which, according to its criteria for the inclusion of data, was primarily, the SMS assessment was primarily based on published academic literature, enabling independent scientific evaluation of IARC's assessment. The agency did not consider the data presented in the Monsanto papers, nor could IARC assess the original data from industry's trade secret studies, with the exception of 4 rodent studies that were sufficiently open and accessible. IARC found limited evidence in humans, sufficient evidence in animals, and strong evidence for supporting evidence for 2 mechanisms of genotoxicity, of cancer-inducing genotoxicity and oxidative stress. IARC found, as we can see, these 2 assessments differ extremely. And since BFR's approach to evaluate the animal evidence has become quite clear, or has been shown quite clear by Peter Clausen before, I will now deal only with the way how BFR has dismissed the human evidence from published studies, of course, and the mechanistic evidence. On what basis did BFR dismiss the human evidence? As illustrated in this slide, the majority of epidemiological studies, also having been shown before by Ms. Gaitan, show increased risk of non-Hodgkin lymphoma in people that worked with glyphosate-based herbicides. And IARC concluded that these human data meet the criteria for limited evidence. That means a causal interpretation is credible, but chance, bias, or confounding can't be excluded. In contrast to IARC's assessment, industry and BFR have concluded that these epidemiological studies were, with one exception, not reliable. But why? Let's have a look. Industry claimed that information on important confounding factor like other pesticides, like smoking behavior, like medical history, would have not been contained in these studies, and therefore these studies should be dismissed. And BFR dismissed the study in its renewal assessment report. However, BFR dramatically, before I come to that, I will just say a reaction of German epidemiologist Eberhard Greiser, who was an invited expert in the Deutsche Bundestag. He just compared the original epidemiological studies with the BFR evaluation, and he wrote in his statement, without a doubt, all the data claimed by the BFR and initially claimed by industry to be missing had actually been ascertained according to good scientific epidemiological methodology. And Professor Greiser also stated that this represents a deliberate falsification of study content, presumable with the intention of qualifying the studies as scientifically inferior. So, let me go further. And after that, BFR, after having evaluated IARC's assessment, dramatically changed its evaluation. Then it suddenly said, BFR agrees in its addendum with IARC that the other IARC categories, that's meaning inadequate evidence or evidence of lack of carcinogenicity, do not fit. But then, in a really remarkable statement, BFR also said, however, BFR adopts a more cautious view since no consistent positive association was observed. I don't understand what they meant. I think it's only to confuse people. Because if there is limited evidence, then it has to be classified as possible cancerogenic. If there is not limited evidence, if there is inadequate evidence, I do not have to classify. So, the next question will be, has BFR been inappropriately been influenced? And this question could be maybe best treated with the genotox issue. As you know, genotoxicity is a basic mechanism and the most problematic one for cancer because there is no safe threshold. And BFR, despite of convincing evidence for genotoxicity in the published literature that led IARC to conclude that there is strong evidence for genotoxicity, proposed to ECHA in its CLH report that no hazard classification of glyphosate for mutagenicity is warranted. And the reason was negative results in the majority of the in vitro and in vivo mutagenicity tests. Indeed, if you look into the ECHA proposal, you will find that BFR has listed 61 genotoxicity tests, and the majority of which, 45, report negative results, and only 16 report positive results. But when you have a closer look on the authors and sponsors of the studies, then you find that those 45 tests that report absence of DNA damaging effects consists of 42 tests that say glyphosate is not genotoxic and from industry, and 3 tests from published studies. On the other hand, all 16 experiments that report positive genotoxic effects in the CLH proposal come from published studies. In other words, 100% of the secret industry experiments tell us that glyphosate does not damage DNA. But when we look at the experiments from independent published studies in the ECHA report, 84% tell us that glyphosate is damaging DNA. And BFR had no problem with that and confirmed the conclusion of Monsanto that glyphosate is devoid of genotoxicity. But when you try to explain that to everyday people, then you get a problem. But the question was, has this decision, which is of course a questionable decision to say glyphosate is not genotoxic, by the EU bodies, has this been influenced by Monsanto? And this was a question addressed to Bernhard Uhl, head of EFSA, and he said no. He said the decision, the studies under scrutiny, there are 2 studies that are discussed in the Monsanto emails. The studies under scrutiny were not prioritized by EFSA. But this is contrary to what you see when you look into the renewal assessment report, which was also the basis for EFSA's, not only for ECHA's decision. In the chapter, published data on genotoxicity, and this is what it's all about, because genotoxicity data is full with positive findings. We find that the review article from Williams, Gross, and Munro, and some of you might know, this is the article which, according to Monsanto email, has been ghostwritten by Monsanto scientists. And this article not only does introduce this chapter on published science on genotoxicity, this article is, and now listen, unbelievable 30 times cited in only 29 pages of this paragraph on published studies on genotoxicity. And even more, if you look at the court documents from the Monsanto litigation in the US now, because according to these emails, Monsanto tried to find or to develop, because they had something like a genotox hole in the 90s, to develop someone who is comfortable with the genotox profile of glyphosate Roundup and who can be influential with regulators and scientific outreach operations when genotox issues arise. This is just from an email from a Monsanto scientist named William Haydens to other Monsanto scientists, and this was because they had a problem then. The person they found, the candidate was James Parry. James Parry was a renowned British expert on genotoxicity in the 90s. He died in 2010. But Parry obviously didn't develop the way Monsanto would have liked. My read, and this is another email from this litigation protocols, my read is that Parry is not currently such a person, and it would make quite some time and dollar per study to get him there. What had happened? Parry has been, and this you can read in the Litigations Act that are all openly accessible, Parry has been commissioned by Monsanto to draft a genotox paper dealing with the unpublished and the published evidence of glyphosate and genotoxicity. But the Parry paper, which is also part of the court documents, as I already said, turned out into something that was a compilation of published studies that provide credible evidence for glyphosate's genotoxicity and thereby contradicted Monsanto's own unpublished studies. Parry proposed that Monsanto should now perform some studies in order to find out if the positive results in these published scientific studies using other methods could be falsified or reproduced. But Monsanto obviously didn't like this idea because the next email, in the same email, we simply aren't going to do the studies Parry suggests. And we have not made much progress and are currently very vulnerable in this area. This is the reaction to it. And we have time to fix that, but only if we make this a high priority now. So this all was in 1999. And this was the time when the first EU approval process was. And then, 2 years later, it looked as if Monsanto had managed to fix this gene tox hole because a former Monsanto scientist whose job duties are described as registration defense of Monsanto's pesticides in EU member states, his name was Mark Martens, and he was praised then in 2002 that he has developed the data to gain key EU scientific support that the reported genotoxicity of Roundup herbicides was due to secondary consequences unrelated to glyphosate, thereby preventing adverse effects on Roundup business. And these secondary consequences unrelated to glyphosate, we do have them in human studies. They are the main arguments to dismiss them. They are the main arguments to dismiss high excessive toxicity mouse studies. And now it's interesting. This ghost-written Monsanto paper, Williams et al., which has been used to dismiss the published evidence for genotoxicity 30 times in a small chapter, this came out 1 year before Mark Martens got this laudatio, and it also uses exactly these arguments. And so if we conclude last summary is human evidence, animal evidence, and mechanistic evidence. We can say that it would be interesting to have a deeper look on the influence of these EU evaluations of glyphosate and cancer. This is my conclusion. Thank you.
Very well. I'm almost convinced that we definitely will need an inquiry committee. But I must say that that discussion probably will be short because we are a bit late upon our schedule. So Marco Antonio, King's College London, expert on medical and molecular genetics, will say something about Roundup causes non-alcoholic fatty liver diseases, which doesn't sound too good.
Yes, thank you very much, Dr. Poc. And thank you for the invitation to be at this very important meeting. So what I wish to highlight In this presentation, I will summarize the work from my group that we have published in the last couple of years, the last paper being in January this year, and want to highlight in a way that carcinogenesis is not the only thing we need to worry about as a potential toxic outcome from glyphosate. Just to review a few figures, because this feeds into the data that I will show in a few minutes. These are the acceptable regulatory permitted daily intakes, acceptable daily intakes within different territories around the world. In the European Union, Australia, New Zealand, as we've heard, it's 0.3 milligrams per kilogram body weight per day. In China and Russia, it's 1 milligram. And in the United States, it's almost 6 times that of the European Union at 1.75 milligrams per kilogram body weight per day. These are what we're told are safe to ingest on a daily basis, day in, day out, with no health consequences. But the question is, are these exposures really safe? Because there have literally been no studies conducted to actually evaluate whether chronic low-dose exposure, either at these permitted levels or below, are actually safe or not. We actually questioned, back in 2012, myself and others, including Claire Robinson, who's here in the audience, published this critique of BFR data, which took apart the scientific evidence, and we came to the conclusion that at least the European Union acceptable daily intake for glyphosate was at least 3-fold too high and possibly 12-fold too high. So we already back then had suspicions that published data was misinterpreted to arrive at an artificially high acceptable daily intake. And we need to bear in mind that why these toxic outcomes can arise. Glyphosate is known to have multiple toxic effects. It is a patented antibiotic. Before it was even patented as a herbicide, it was patented as a nutrient metal chelator. It will strongly bind manganese, magnesium, zinc and cobalt, among other metal ions, therefore depriving these vital nutrients to the plant and the consumer of those food products, including, of course, removing them from crucial enzymatic reactions. We've heard how glyphosate is a proven DNA mutagen, and there is some, although I would say at this stage, inconclusive evidence that glyphosate may be an endocrine or hormone disruptive chemical. And in addition, as has been touched on by others, we have to remember that glyphosate is never used alone. It is the active ingredient of commercial formulations with other substances collectively known as adjuvants, the nature of which are a closely guarded industry secrets. You never actually know exactly what is in a given formulation. But in studies conducted by my postdoctoral fellow before he joined me, Robin Menage, he consistently found in his in vitro studies that commercial formulations were more toxic than the active ingredient alone at certain concentrations. And I mentioned this here twofold. Firstly, I'm going to show you data with Roundup formulation, not with glyphosate alone, because this is what we are exposed to. We're not just exposed to glyphosate alone. A real-world exposure is to a glyphosate-based formulation. And although at European Union level, at said EFSA, only the active ingredient of pesticides is actually evaluated for toxicity, it is up to individual member states to evaluate the toxicity of formulations. So our study feeds directly into what individual member states actually have to consider. In terms of how much glyphosate is in the general human population, there have been a limited number of surveys in Europe and North America. I don't expect you to see all these little numbers in this table from the Neiman et al. Paper from 2015, but the conclusion of this paper is basically that the daily intake is a fraction of the permitted acceptable daily intake, and therefore there is nothing to worry about, even though the surveys that have been conducted find glyphosate present widely within the human population, about 50% in Europe and virtually 100% in the United States. The question arises is, Are the levels detected in humans, which are below regulatory set limits, are they safe? Because as I mentioned earlier, nobody has actually evaluated the consequences of chronic low-dose exposure to glyphosate-based herbicide. So what's needed is a long-term laboratory animal toxicity feeding studies at or below the acceptable daily intake to see are these truly safe as the regulator, as regulatory agencies around the world purport, or whether they can actually give rise to negative health outcomes. And I begin by, this is the beginning of the research in my group. It's based, we used materials from this study led by Professor Gilles-Éric Seralini, which many of you may be familiar with, and some of you may accept the fact that it was discredited and had to be withdrawn and republished. But apart from the fact that the reasons given for its withdrawal are totally spurious and invalid, the material that we used in our investigations from this study have never been, the validity of this material has never been questioned. And so what we've done clearly stands independent from this study as well. What we focused on was using tissues from from this study, the groups of animals that were fed Roundup in their drinking water. So, and they were fed the Roundup diluted in the drinking water over 24 consecutive months. And there were 3 doses used, as you can see in the lower part of this slide. And the dose that, oops, what have I done? Sorry about that. That's just to make sure you're staying awake, just to... There we go. Sorry about that. Okay, I pushed the wrong button. So we focused our investigation on tissues from rats that were fed this very low dose of Roundup in their drinking water. The dilution of the Roundup gave a final glyphosate equivalent concentration of 50 nanograms per litre, or 50 parts per trillion. And in terms of a daily dose, daily ingestion, it was only 4 nanograms per kilogram body weight per day. Just to put that into perspective of the acceptable daily intakes, these rats ingested the glyphosate equivalent of Roundup, which is 75,000 times below the EU acceptable daily intake, and 437,500 times below the US acceptable daily intake. So in other words, incredibly low doses, but reflective of a real-world ingestion, what you and I would consume in what is permitted in drinking water in the European Union. In the original Seralini study, the findings in these animals that ingested this lowest dose of Roundup was that they had 3 times more pathologies observed at an anatomical organ level by microscopic analysis. Their blood-urine biochemistry suggested impairment of liver and especially kidney function. There were also disturbances in the hormone balance in these animals. Testosterone levels increased by 96%, whereas estrogen dropped by 26%. And that the electron microscopic analysis of the livers showed statistically significant alterations in the cell nuclear structure, suggesting major changes in gene function. So even in these early observations, there was good signs that even this ultra-low dose, really low dose of Roundup ingestion was causing problems in these animals. So what we did was in order to confirm and extend these observations, we took the organs from these animals and in my laboratory we conducted molecular profiling, or what is known as omics analysis. This consists of a comprehensive in-depth gene function and molecular composition analysis. It's highly sensitive, it's highly predictive or indicative of health or disease status. And what I mean by these omics analyses are transcriptomics, which looks at the patterns of gene function profile, proteomics, which is the protein content of the organ, and metabolomics, which are the small molecule metabolite profiles of these organs. These are tools that are now revolutionizing the way we do our analysis in science, including in clinical practice. So what we did was we took from the female animals that that ingested this lowest dose of Roundup, this 4 nanograms per kilogram body weight per day. We took the livers and the kidneys from these animals, bearing in mind that the amount of Roundup in the drinking water was half of what would be permitted in European Union drinking water in terms of glyphosate equivalent. And this we published back in 2015, the paper which you can download from Environmental Health Journal website. Transcriptome profile analysis reflects rat liver and kidney damage following chronic ultra-low dose Roundup exposure. Just to quickly go through some of this data, in this what is known as a principal component analysis, basically the results of the 28,000 genes, the levels of function of the 28,000 genes in the livers and the kidneys of these animals, in each individual animal, the 10 treated, which are the red, and the 10 non-treated, which are the green, are basically projected in three-dimensional space. And the message I want you to take away from this slide is that the group of animals that are treated, the red spheres in both the liver and the kidney, are separate very clearly from the green spheres, which are the control group. So already this is telling us that the effects of this ultra-low dose Roundup is having a differential effect in the gene expression profile in these two organs in these animals. And when we look at the levels of alteration, we found that in both the livers and the kidneys, more than 4,000 genes were either decreased or increased to a highly statistically significant level in either the kidneys or the livers of these animals. These were levels that, if you look at the table in the lower right of this slide, it gives you an idea that at the level of statistical significance that we set at 0.01%, by chance we would expect only 380 genes to be altered, whereas what we found in reality was more than 4,000 genes either up or down in their levels compared to the controls. So highly significant changes. And out of these more than 4,000 genes, 1,319 were common to both liver and kidneys, changing up or down in the same way. And when we looked at these more than 1,300 genes and scrutinized them in what's known as the gene ontology database to find out what are the consequences, what are these changes in gene profiles reflective of, what are they telling us about the health status of these organs, what we basically found was that these organs were damaged. They were reflective of necrosis, fibrosis, ischemia, and oxidative stress. So basically, these gene expression profiles were predicting that these organs were not healthy. They, in fact, were highly unhealthy. But the gene expression is a predictive measure. Not all changes that you see at the level of gene function end up reflecting changes in the products of genes with health consequences. So we wanted to go further than just looking at the gene expression pattern. And so we took the same livers, the same liver samples, and subjected them to now an analysis of the protein content and the metabolite, small molecule metabolite content, the proteomics and the metabolomics. So unlike the gene expression, which predicts, the proteomics and the metabolomics actually measures the physical composition of the organ. And that will tell you, be reflective of the health status of the organ. Either it's going to reflect health or it's going to reflect disease. And we published this analysis back in January in Scientific Reports. Again, you can download this from the journal website. The title of our paper, Multiomics Reveal Non-Alcoholic Fatty Liver Disease in Rats Following Chronic Exposure to an Ultra-Low Dose of Roundup Herbicide. And what we found in these rats, first of all, when analyzing the triglyceride fat levels in rats, in these animals, we found that at later stages in the 2-year period of ingestion, we found that there were statistically significant elevated levels of fat triglycerides in the blood of these animals. This is important because elevated triglyceride fat in the blood is a risk factor for non-alcoholic fatty liver disease. And however, turning to the proteomics and the metabolomics, again looking at this principal component analysis, we found that with the proteomics that you see on the left, we see, again, a clear separation of the control and the treatment groups, although with the metabolomics there was a mixture, there wasn't a clear separation, but still statistically significant differences in many metabolites. So in the proteome alteration reflects a liver lipotoxic condition out of 1,906 proteins that were detected. 214 were found to be altered, and they reflected a fatty liver toxic condition. And the metabolite changes similarly. Out of 673 metabolites, 55 were highly statistically significantly altered, again reflective of lipotoxic and oxidative stress, and in other words, there were markers of liver, elevated levels in markers of liver toxicity. So our conclusions from the rat liver proteome and metabolome are that they revealed substantial overlap with hallmarks or biomarkers of non-alcoholic fatty liver disease and its progression to severe fatty liver disease known as non-alcoholic steatohepatosis, and the liver functional dysfunction resulting from chronic ultra-low-dose glyphosate-based herbicide exposure. And just wondering, why is NAFLD and NASH so important? Is because it's seen as the new plague of the 21st century. I don't have time to go into this slide in detail, but it really is affecting a very high percentage of US and European citizens. And not only are the symptoms of this disease severe, but also they result in a marked heightened risk of liver cancer as well. Hence, these findings are relevant to the current controversy surrounding whether glyphosate is linked with cancer or not. And what we believe we have found is what our data is indicating is that glyphosate-based herbicide exposure constitutes a new risk factor for severe fatty liver disease. And in this position statement that we published last year, the conclusion, what I hope the data I've shown has shown you is why the position we arrived at was that regulatory estimates of tolerable daily intakes for glyphosate in the United States and European Union are based on outmoded science, and that you can have toxic effects after chronic low-dose exposure, and that currently the safe daily intake of glyphosate is currently unknown. And while that is unknown, there is only one sensible way of regulating it, which is to basically withdraw it from the market until we do know what the safe level of ingestion is. Thank you very much.
Thank you very much. It looks like we will, or our agencies will need this current contemporary science and not only feed rats with something or look at it and if it die, it's wrong. So thank you for this presentation. And our last speaker is Hartmut Frank from University of Bayreuth, and he will elaborate, do chemists scientists need to understand ethics? I would like to know. Frank.
Mr. Chairman, thank you very much for the invitation to come here to speak to you with this sentence, do chemists, scientists need to understand ethics? I think there are a few presentations in the few hours behind us where we can, so to say, just erase the question mark and can state that they do need it. I just show you where I come from Bayreuth, just to give you these 5 minutes because we don't have much more. An idea of how we get here to this situation. Why do we ask it? Personally, I'm a chemist trained in toxicology, and then when I became more involved over 30 years working with toxicologists, this question rose up in me when observing my colleagues how they treated their data. That's the reason why later on I became interested in the question of ethics. A conflict of interest, a conflict I do not have except the conflict with these gentlemen, because I'm not an ardent lover of, let's say, Teutonic operas. I have the same position like Mark Twain. who said when he visited Bayreuth, who is known for its Wagner Festival, opera festival season, I have been told that Wagner's music is better than it sounds. Maybe this applies to some of the literature we read. So therefore, I think just let's spend a few minutes. Chemistry is certainly something which has its standards and based deeply on philosophical grounds of the age of the Enlightenment. We always use the powerful foundation of science in order to come to these deliberations as we have them here. The typical scientific approach is falsification of a hypothesis. We ask and we answer this hypothesis with yes or no. So this is in principle a very exact approach, and we are very proud of it because we know the whole truth. When we look, however, a little closer, a hypothesis is just conceived by a scientist who has his prejudices and his points of views, and what we only do is we falsify or we cannot falsify the hypothesis. So in principle it's not yes or no, that's very important. It's not no or no. That's how we answer scientific questions. Oh, sorry, it's backward. Now, when I looked at the glyphosate molecule, I had been working as a chemist a bit with glufosinolate. At that time, it was about the same time when glyphosate and glufosinolate came onto the market. I worked a little with the analytical approaches. And I still remember that at that time these two were competing. Unfortunately, you cannot read the explanations to the right of these formula, but I immediately was somehow reminded of thalidomide. Namely, thalidomide, the famous teratogenic agent introduced in the late 50s and early 60s, is not immediately visible that it is similar, but when you look closer, it is quite similar because it contains glutamate. It's known that thalidomide in the body at pH 7.4 within about 5 hours, that might be the residence time or even longer, is 50% hydrolyzed to glutamate. Now, glyphosate, and for both glyphosate and glufosinolate are reports that they inhibit glutamate metabolism. Glutamate is an excitatory neuroactive amino acid, and neurotransmitter, that's what I wanted to say, a neurotransmitter. And so it's clear, and it's also just recently discovered by some groups, that it also a signaling factor between an intercellular communication. So it could very well be, and it's in my opinion most likely, that an important part of the complex physiology of these pesticides and these agents is an interference with glutamate metabolism. This is, however, circumstantial evidence. It's not fully clear, but I'm surprised that although there are just in recent times more and more reports, it has just been mentioned in a sentence by the preceding speaker, that very little has been spent in looking at this possibility. Especially in a country, I just had the chance in 2013 visit some with my colleague, my friend, Franciscan priest, the man in the green, second from the left, several families. He takes care of bodily handicapped people, and he visits them. And I was surprised how many are there in these soybean growing areas. They are like prairies of soybeans. In these soybean growing areas, how many people are similar to our thalidomide cases, although they are much younger. They cannot be due to thalidomide because they are too young. They are not 50, they are 30, 40. And at that time, thalidomide was from the market. And it's really surprising, although that's again circumstantial evidence, that there are quite a considerable number. And these are the people, not so many in Europe, these are the people who are heavily exposed, the farm workers in these large haciendas. And I think there we have a problem. What is the problem? First of all, it's a quantitative one. As you can see, unfortunately I don't have a pointer. In 1974, glyphosate was introduced. At that time I was a postdoc. And this was zero. Until the following 20 years, it rose to about 60,000 tons globally. And with the next following 20 years, it rose from 60,000 tons to 800,000 tons. And that is where the story begins. We are starting the true global experiment now. The dose is rising. It's typical like in toxicology, like it has been told, and that's the principle I often observe when people talk about risks. They talk about substances, but very little about doses. Certainly it is happening, and it is certainly done under checking the publications. But here, when we look at the epidemiological data or observations, we have to take into consideration that it takes a long time from the beginning until we see the outcome, especially in carcinogenicity and teratogenicity. That is something which we perhaps start to see in the newest literature, not the one which has been published 20 years ago. And secondly, we have a qualitative one. As a result of coldly materialistic, scientific, economic, non-empathic culture of science. That's what I would state here, which is true when you look at the dose effect in a cell culture experiment or in a rat experiment. But when we look at chemicals which are spread to many people in relatively unprotected areas, then we have to adapt something else. We cannot insist that science holds the whole truth and exact truth, caring little about fairness, especially as we know that there is not a yes and no answer, but a not no or no answer, and that it is not true that science is the absolute truth. It is just the preconceived ideas which are either proven or disproven. But we never go into the objective, balanced assessment of the problem and risk associated with a chemical. Daniel Zarewicz, some years ago, wrote an interesting article or published an interesting article in Environmental Science and Policy entitled, now scientists must listen really carefully, How Science Makes Environmental Controversies Worse. And it's very enlightening to read that article. He says, scientific uncertainty, which so often occupies a central place in environmental controversies, can be understood not as a lack of scientific understanding, but as a lack of coherence among competing scientific understandings amplified by the various political, cultural and institutional contexts within which science is carried out. And another eminent physicist, Christine Schrader-Fischette, who also holds a chair in biophysics and ethics, says, it's also an interesting article published in Environmental Justice, Participation and the Law, analyzing public participation in risk analysis, how the wolves of environmental injustice hide in the sheep's clothing of science. I mean, I could actually finish here, but I think it's really important that we think how can we change the situation, because I think sustainability is equivalent in my mind to an integration of ethics and science. What is needed? I think for the next generations of scientists, we need to introduce them into a better understanding that science has its limits, and that we also have to, when we come to applied science, when it has consequences on human beings, that we also have to foster their empathic abilities to understand its limits. That means concretely educating in applied science ethics in curricula. I mean, for instance, Edgar Morin published a paper where he gives such possibilities by UNESCO, and I think this is a good guideline how we can approach such a problem, and in which we are sticking now. In conclusion, I would say a new paradigm in science teaching has to be developed, integrating scientific, intellectual, analytical and philosophical, ethical aspects. A higher level, the strong emphasis of international and interdisciplinary cooperation needs to be achieved. I would like to stop here. I think I have made my point clear, but I believe that we have to do more, especially the stakeholders, like in a meeting like this, have to be introduced, like, for instance, this family from Brazil you have seen. They should also be brought on stage, and they should give their opinion. Thank you very much.
Thank you very much. You made it in your 15 minutes. Very good. Thank you. And for a short discussion, I would like to ask Kate if you could join us, and Tim, and that's it. Very well then, we have something like 15 minutes for discussions. I have two people. Yes, Stuart, you are first. Then Martin Pigeon. Yeah, it's you. Very well. And then next is okay. So we will start with you, Stuart, please.
I think the reason why there's been such a big interest in glyphosate isn't because the ground has suddenly stopped growing things. It isn't because there's suddenly been a great rash of deaths concerned with glyphosate. It is because the Green lobby have been dismayed that GM crops haven't killed everybody, that they're steadily gaining ground, and they cannot prove that GM crops are harmful, so they've gone round the other side and say, GM crops are associated with glyphosate, let's try and find something wrong with glyphosate. And as soon as you try and find something wrong with it, with something synthetically made by man, of course you will. And I remember pre-glyphosate days. I am 6 or 7 years old, it came in 1977. Before that, we had to cultivate the ground regularly to kill perennial weeds. That meant that we exposed the soil to seagulls who ate all the earthworms that were disturbed by the cultivations. We created problems to the soil. And, of course, we burnt up fuel, pumped all this CO2 into the air, which people say is a terrible crime. And if the weather went against us, the whole thing was absolutely useless. There is a cohort of people now who, for many years, have been exposed far, far more than anybody else to glyphosate, and that is people like me who sprayed it for 20 years. And those who worked in forestry, gardeners who didn't take all the precautions. Often you got soaked in the stuff for using a knapsack sprayer. Now, our generation, those of us who've done that, we should be coming forward now in doctor surgeries and they should be waving red flags, but they're not. We're told that it causes cancer, and it's been listed in this block of things that cause cancer, which include working night shifts, sitting in front of a log fire, contact with dry cleaning fluid, cutting human hair, and drinking mate, the Argentinian national drink. That is, glyphosate has been put in that bracket of carcinogenic materials. What is even worse, though, are alcoholic drinks, hormonal contraceptives, and processed meats. So if you're going to ban glyphosate, you've got to ban all that lot as well. Otherwise, it simply isn't logical. As far as toxicity goes, the organic farmers use a material called rotenone that's far, far, far more toxic than glyphosate ever was, and I've been smothered in glyphosate over my life. Where's the rash? Where's the leper? Two final points. Somebody said that the seeds of GM crops can't be resown. That was the case for 2 or 3 years. And the Green Lobby said, oh, what about the Terminator gene? So now, if you want to resow GM seeds, you can, but you must pay the breeders a levy, as you must do with non-GM seeds. And finally, we all got rather excited as farmers about glyphosate being a rat killer. We wanted something that was really good, and we played around, we tried it, and it's absolutely useless. Thank you.
Very well then, but I'm afraid that I didn't quote a question. Maybe it's because I'm not native. It's maybe because I'm not native speaker, but I think that we have here Professor Antonio from King's College of London. Maybe you caught as a native speaker the question in this.
Birth to children, for instance.
I don't know. Of course, this was a sort of attempt to make sort of scientific analysis of possible harm from glyphosate. I don't know. Could you, as a native speaker from England, could you react maybe to the, English remark?
The point is that we know that chronic illnesses in society, in industrialized nations, Europe, especially North America, have escalated enormously in the last few decades, and especially in the last 20, 30 years. And so something has changed during these recent decades that have resulted in the escalation of these chronic illnesses. Just to highlight one, which, cancer has been a theme here. We now, at least in the UK, is probably the same here on the continent, that it's expected that roughly 1 in 2 men will get cancer and 1 in 3 women will get cancer. Same in the United States. Why is that happening? It never used to be the case. And this is across all age groups. Something has changed in that time to result in this massive escalation in chronic illnesses. When you ask what has changed, many things have changed. But one of the things that has changed enormously, which affects us all since the Second World War, has been the way we grow and consume our food. It's become a chemical-based agriculture. And the consequences of this, I believe, are being underestimated. Research like ours is beginning to highlight that, given enough time, even what is considered safe to be exposed to, and we've only looked at one. We're talking about we're exposed to cocktails of tens, if not hundreds of different chemicals from different sources. Work like ours is beginning to highlight that what is considered to be safe in the past may not be the case, and that we need to revisit how we look at, how we evaluate toxicity, so that it's in line with the latest scientific understanding and methodology. And to go out there and look for the associations. This gentleman here behind me, he may not know, is that actually we're not looking in the human populations properly to see if we can find associations with different cocktails of chemical exposures, especially pesticide exposures, because that's what we're ingesting day after day, and different disease categories. And until we have that linked with controlled laboratory animal feeding studies where you can isolate effects like what we've tried to do, you have a controlled lab environment where you can isolate a compound and its effects and derive a direct causative relationship that you can then correlate with an epidemiological observation. Exactly what IARC has been doing, of course, in its cancer evaluation of glyphosate and many other substances. This is what we need more of, and why we can't, there are too many unanswered questions, and we need to move forward cautiously as a result.
Thank you for this reaction, and Stuart, I promise you next time I will organize something about your favorite GMOs. This was not about GMOs, I don't care about it too much. Next was from behind, I think it's Martin Bijan, yeah?
Yes, thank you very much. I have one brief statement and two questions. My name is Martin Pigeon. I'm working with Corporate Europe Observatory. I would like to make a brief statement on behalf of the European Citizens Initiative Stop Glyphosate, which has gathered more than 733,000 signatures as we speak, which makes it the fastest ECI ever done at the EU level, which shows that this issue goes way beyond whatever the green lobby would be able to put out. This shows that citizens are there too, that they want and can have an impact on this decision. This European Citizens Initiative revolves around three demands. The first one is to ban glyphosate because of the considerable environmental damage that has been documented, as well as the high probability that we've heard today that it would cause cancer to humans, in particular to farm workers. That's the first demand. The second demand, I think, goes across many of the discussions that we've heard today about the need to have all pesticides be evaluated on the basis of published scientific evidence only, and not mainly, as seems to be the case right now for EFSA and ECHA, data provided by industry which cannot be assessed by any independent scientist because it is secret. It's not published. The third demand is to tie the glyphosate issue to the broader issue that was just mentioned right now, actually, to the fact that chemical alternatives that are just as toxic and possibly even more, and probably more expensive as well than glyphosate, are available, and that if you don't take a bit of a bigger picture and only ban glyphosate, you might not necessarily improve things overnight. So the third demand is about the EU committing to reduce pesticide use across the board and support farmers in this transition, because again, farmers are the first in the line in this. This was my statement. I have two very specific questions. The first one to Tim Baumer from ECHA, which is, how do you explain such a strong difference as far as the genotoxicity is compared to the work of IARC? And I would second the question by Peter Klausing at the end of his presentation. Why not even considering the category 2 classification? And my second question is to Peter Klausing. Did I understand correctly that the reason why the 2001 study was discarded by EFSA or ECHA, I wasn't sure, was because of the intervention via phone conference of Jess Rowlands from the EPA? Thank you.
Thank you. Closer, yeah, very, very well done. So please, we have 5 minutes before we are kicked from this room. So.
Okay, thanks for the question. I'll try and answer the one in relation to IARC and how the differences occur. I think, as you heard from Kate's and my presentation, the methodology is quite different. If you depend on studies in the public domain, you get a certain data set, and there are advantages to that, as Kate quite clearly said. If you have access to a much more broader set of studies, which we did at ECHA, we had full access to the original studies, you are able to get an appreciation of the original laboratory information, unfiltered by, let's say, what other people have said, which is also in the public domain. And that's really what we attempted to do in our evaluation of all of these studies, is to dig as deep as possible with the original information rather than be reliant on who said what at which time. I have no dispute, and ECHA has no dispute with IARC whatsoever. We fully understand their position and their way of doing things. We have an obligation under our regulations, which we work under, to do them in a different way. That's all I would like to say on that. As to why we didn't go to category 2, we did not see there was enough evidence to do so. If you think about it, it would have been an easy way out for committee members faced with a difficult decision to go down the middle. They chose not to. Thank you.
Yes, this is the only traceable fact which caused to kick out the 2001 study, as it was even confirmed last week by Jose Tarazona. And I would like to use the opportunity to point out that Kate Guyton said in her presentation that observers do not interfere with the deliberations of the expert committee at IARC. In case of the EFSA, we have clear evidence that an observer and the EPA representative, who actually is mentioned explicitly in the Monsanto emails which became public, has been interfering, and he has been heard by the committee or by the group of experts. So this is quite bothering me.
Mr. Chairman, thank you for the opportunity to say a few words. I felt I had to say a few words.
Just introduce yourself.
I will. My name is Eiras Jones. I work for the European Crop Protection Association. I'm representing the industry and obviously very involved in this issue. It's actually quite difficult to ask a question because there's a lot of issues that have been raised. There's a couple of points I just wanted to pick up on. One is something that Mr. Coggins said early on about safe use. Yes, it is about safe use, about proper use of our products. And what we want is to minimise the use of pesticides. But minimisation is actually not banning a few. Minimisation is actually having the tools available so that we can minimise the use of a wide range of tools out there. We need the tools to be able to minimise the actual use. It's a key point. Mr. Coggins suggested that actually glyphosate allowed more monoculture so that we can actually grow the same crop every year. Actually, that's a little bit, it's not untrue, but it doesn't mean that you have to go with the same crop every year. It also allows you to go in with a different crop every year. So there's no link to monoculture and the use of glyphosate. There's a very clear link between glyphosate and the protection of earthworms, because the fact that we don't plough means that we save a hell of a lot of earthworms and keep the soil structure in place. Another important point. There was a point here, and I understand Mr. Agnew didn't ask a question, but there was a point that was made in response about we don't know why cancer has gone up. Actually, I think most people would tell you we do know why cancer has gone up. We're actually living longer. And there's a key point. And there's a key point if you look at...
Nice statement, but could you come to question, please? I'm sorry, we are...
Well, I think that is a key point. Maybe a question to the gentleman at the front. So saying we're looking at a cocktail of chemicals, and we should look at especially pesticides. Actually, I think there's a question here. Why are we looking especially at pesticides? If we're interested in cocktails, there's a cocktail of chemicals out there as a whole, and we should be looking at the big picture of all our chemical exposure. We shouldn't be focusing on a small group. We need to understand everything. The amount of time and studies and money we've spent on one chemicals when we should be actually looking at everything is completely disproportionate. So the question is, why focus on one small section?
To whom?
I think to the gentleman, with the English gentleman at the front with a Greek name.
Very well. Good news from you is that you want to sell less products. It's good news. And
you're absolutely right. We do need to look at not just the cocktail of pesticides, but I would challenge you to say that is just a small group. There are hundreds of approved pesticides that are being used routinely, And therefore, we are exposed to many tens, if not hundreds, of pesticides through the food chain. And therefore, they represent a very substantial exposure of substances in the human population. And it's something that is virtually unavoidable. It's something that everybody is exposed to through their food every day, which is why they constitute a very important subclass of chemical exposures in the human population. So that's really why I feel we need to focus on them. And your point about the reason why there is more cancer because we're living longer, I'm afraid that is totally inaccurate. The increases in cancer are across all age groups and most worryingly amongst children.
You can say.
Yeah, the main reason is it's the biggest sold chemical on Earth nowadays, close to 1 million tons annually. I mean, I remember the times when we were talking about another, let's say, environmental toxin like tetrachloroethylene is also one simple compound. Why should we concentrate on that when it was, so to say, regulated down? Because it was produced in such tremendous quantities that it caused problems. And I think the same is true for glyphosate. As soon as you reach 1 million tons annually, you have to talk about it. You cannot just say it is one of those pesticides and why do you concentrate on those? I mean, this is simply does contradict basic toxicological thinking if you just go at this in this way. I mean, and also cancer is clearly now every epidemiologist, starting with the old Doll and Peto study, clearly always age correlate the incidence of cancer and try to get this confounder out of the picture. I mean, they are not stupid nowadays anymore. They have made mistakes and made some mistakes in statistics and epidemiology in the 70s and 80s, but these times are over.
Very well, thanks. And I've seen a last, absolutely last question or remark, and be brief, please, because we are overlapping time.
Thank you. It's for Professor Baumer. Recently, I reviewed the Glyphosate Task Force updated literature search for endocrine disrupting effects of glyphosate. It was only for about a 3-year period they were updating a literature review. And they said when they searched PubMed, they found 7 potentially relevant studies. And that seemed a little bit low to me. In a few minutes, I found 4 more that the literature review, no matter the other databases that they also searched. So they missed a good fraction just of the published literature. But my question, related to that, my question is, relevance and reliability are being totally confounded and messed up in evaluations. So I'd like your reaction on this. Relevance is for the initial assessment of the abstracts. You're looking at the abstracts and the titles, and if it's at all relevant, you're supposed to go on to evaluate it. And this is what I found in this literature review, and I've found it everywhere else in pesticide RARs. Your reaction?
Well, you started your question about reproduction and about, I think, endocrine disruption. Part of the classification and labeling proposal from Germany was also for reproductive toxicity. And again, as with carcinogenicity, there were a massive amount of long-term animal studies involved in that, which were also gone through in the same way in great detail, again to original reports where possible. So reproduction as a whole has been looked at, in our case, as thoroughly as has carcinogenicity. As to your comment on the relevance of, I think you're referring to the non-guideline studies in the open literature. Yeah. Some of those are relevant and are taken into account, but some of them are not. And it's always going to be in some way a selective process with that amount of data. I'm sorry.
Okay, very well then. Thank you all. We have to finish because another group is waiting for the room. I would like to thank you all for being here. Thank to Mrs. Pilar Goya from EuCheMS, Pavel Drashar, UCT Prague, Tim Bomer, ECHA, Sylvia Lacorte, CSIC, John Coggins University of Glasgow, Catherine Gutton from IARC, Tor Midwet, Karolinska Institute, Peter Klausing, Pan Germany, Helmut Brechtschaden from Global 2000, Michael Antoniou from King's College, thank you very much, Hartmut Frank, University of Bayern. Thank you all for being here, thank you for your lectures, and I hope to see you again, because I am convinced that this is not a last discussion about glyphosate in the premises of European Parliament. Thank you very much.
Thank you very much. It was nice meeting you.